It's raining there right now. Yeah, it's raining that side, right? Yeah. I think we are live on YouTube now.
So I think we still have 12 minutes to go. Shall I start the webinar?
It's raining there right now.
Let us wait for the judges to join. No, Tarun, please.
I think we.
The ads would run till that time. Is it okay?
Okay.
Huh?
Because I am only little worried that we should be able to catch ki ye log aaye hai.
Yeah. So we can do we can just mute the. Good evening. Good morning, sir. Vinod Shah, sir.
Yes,
Good morning,
good
sir.
morning, good evening, good afternoon.
Good morning, sir. Good morning.
Okay, I'll start the ads. And
Okay. Then we'll keep quiet.
Insulin, cosmetics, running shoes. These products could not have been made if not for BASF Antwerp. It's the second largest production site of German chemical concern BASF. Our 50 production plants make essential raw materials for virtually all processing sectors, from pharmaceuticals and hygiene products to the automotive industry. The site of BASF Antwerp is about the size of Antwerp's city centre. It's located at the heart of Europe and well connected to rail, road, waterways and pipeline networks. Our key operating principle is Verbund. All our plants are interconnected, meaning that the end products of one production plant serve as the raw materials for another. This way we generate less waste and need less transport and energy. For example, to make diapers superabsorbent, boats supply our steam cracker plant with naphtha as a material, which is then converted into propylene. Propylene is then processed into acrylic acid material for superabsorber used in diapers. Ensuring a safe working environment for employees and neighbours is priority number one. Every day, our more than 3,000 employees combine their expertise, knowledge, drive and creativity to make sure our plants run safely and smoothly. At BASF, we keep innovating for a sustainable future. We create chemistry. insulation, cosmetics, diapers, running shoes. These products could not have been made if not for BASF Antwerp. It's the second largest production site of German chemical concern BASF. Our 50 production plants make essential raw materials for virtually all processing sectors, from pharmaceuticals and hygiene products to the automotive industry. The site of BASF Antwerp is about the size of Antwerp's city centre. It's located at the heart of Europe and well connected to rail, road, waterways and pipeline networks. Our key operating principle is Verbund. All our plants are interconnected, meaning that the end products of one production plant serve as the raw materials for another. This way we generate less waste and need less transport and energy. For example, to make diapers superabsorbent, boats supply our steam cracker plant with naphtha as a raw material, which is then converted into propylene. Propylene is then processed into acrylic acid. Acrylic acid is the raw material for superabsorber used in diapers. Ensuring a safe working environment for employees and neighbours is priority number one. Every day, our more than 3,000 employees combine their expertise, knowledge, drive and creativity to make sure our plants run safely and smoothly. At BASF, we keep innovating for a sustainable future. We create chemistry.
Hello, good morning, good afternoon. Is anyone speaking? Because I don't hear anything.
Yeah,
No,
hi.
nobody's speaking.
Okay.
Only the ad stopped.
Yeah.
Okay. Yeah,
So
I just
anyway,
wanted to
we'll-
check. I just wanted to check.
Yeah, so maybe we say good morning, good evening, good afternoon to everybody.
Yes.
Hello.
Absolutely.
Hello. Good afternoon, good evening, good morning.
Hello, everybody.
Hello, Dr. Barton. Good afternoon.
Good afternoon. Nice to meet you all.
Hello, Dr. Barton, Dr. Shah.
Hello.
Hello. Good morning, good afternoon, good
Dr.
evening.
Sandeep Tiwari is joining in 2 minutes, he is told.
Okay. In the meantime, Tarun,
Yes, sir.
we can finish our 5 minutes before he joins, no?
Okay.
Yeah.
Because time
but,
today.
We can start only at 5, no?
2 minutes
It's-
before
2 minutes
5.
more.
2 minutes. 2 minutes
Yeah.
before 5 we'll start. Because today the time aura is not good after 5. 2 minutes before.
Okay, so
And
I think we,
that 2 minutes I will only take, so it will not affect anyone else.
so we need to start right away. It's just
Right
2:05.
away.
Yes, Tarun, you can start, Tarun.
It's already 4:58.
So you want to address to begin and then I start with the presentations?
Yeah,
No,
but,
I will start, no, as per the agenda.
Opening remarks by you, sir.
Yes. So can I start then? Tarun,
Yes,
are you
sir.
okay? Can I start?
Yeah, you can start. Sir. Yes.
Okay. Hello, everyone. Say good morning, good afternoon, good evening to all the judges, all the DRPI leaders, SPDS leaders, the research students, research presenters, their guides, industry participants, and all sponsors and others. Today, as we stand on the precipice of another incredible year for the Dissolution Research Presentation International, DRPI, I am filled with profound joy and gratitude. It is both an honor and a privilege for me to serve as the General Secretary of Society for Pharmaceutical Dissolution Science, SPDS, and to have been a part of enduring journey that DRPI year after year. Our journey began with a simple idea ignited by the visionary of Professor Saranjit Singh, the global chair of DRPI today, to create a platform for budding research students. The vision was wholeheartedly supported by eminent academic leaders such as Professor Mala Menon, Dr. Krishnapriya Mohan Raj, Dr. Hema Nair, Dr. Varsha Pradhan, who played pivotal roles in the central committee. Further, we are immensely grateful to the dedication of Dr. Farhan Ahmad, Professor Vishwajit Mukherjee, Professor Vandana Patravale, Professor Narayana Brima Udupa, and Professor Swarnalata Saraf, who served as chairs in various zones across India. Behind the scenes, a scientific committee comprising over 60 experienced and esteemed pharmacy educators from diverse institutes and universities in India formed the backbone of this unique program. At the heart of our endeavor lies a deep commitment to nurturing the next generation of scientists, the academics. Under the visionary leadership of Dr. Saranjit Singh, Dr. Mala Menon, Dr. Krishna Priya, we launched the innovative DRPI competition, extending to support to MPharm, PhD, and young industry researchers. The initiative was gathered tremendous momentum as on today, and this year we are thrilled to have DRPI Disor Research Competition International to a global stage. The Society for Pharmaceutical Dissolution Science, SPDS, established as a charitable trust in 2012, has consistently championed the advancement and application of dissolution science worldwide. Today, SPDS proudly stands at the global society as a sole global society dedicated to this noble objective, contributing significantly to the quality of pharmaceutical products in collaboration with the American Association of Pharmaceutical Scientists, that's AAPS. We express our deepest gratitude to Dr. Vinod P. Shah from the USA, whose instrumental efforts helped expand SPDS globally and forge this invaluable collaboration with AAPS. So once again, thank you, Dr. Shah, for this. DISO India, an international conference on dissolution science and its applications, held annually in India, and DRPI, a distinguished research presentation competition for masters and PhD students worldwide, are the flagship scientific events of SPDS. We are also delighted to have partnered with APTI, Association of Pharmaceutical Teachers of India, to conduct DRPI annually. To all the brilliant research students participating in DRPI 2023, I assure you that your journey through this research presentation competition will be both enriching and enlightening. My heartfelt thanks extend to all the delegates who have registered, joined, and participated from all corners of the globe. And it is your collective spirit that fuels this competition. Moreover, I want to acknowledge and appreciate the unwavering support of our partners. This global competition would not have been possible without your invaluable contribution from SOTEX, ACG, and BASF. I must take a moment to recognize the outstanding commitment of our esteemed office bearers, Professor Padma Devarajan, our current president, former president Mr. Vijay Sirsagar, Vice President Dr. Rajiv, current scientific chair Dr. Munira Momin, former scientific chair Dr. A.K. Bansal, Dr. Padma Devarajan, and Dr. Mangal Nagasangar. Special thanks due to our IT expert to Mr. Tarun Soni of NIC Interactive. As we embark on the exciting journey of DRPI 2023, I extend my warmest congratulations to all participants and today's winners. I invite you to share this remarkable event with your friends and mentors in other universities so that together we can inspire even more contestants for DRPI 2024, doubling our impact in the coming year. In closing, I wish all of you a splendid experience of DRPI 2023 World Finals filled with learning, inspiration, and camaraderie. Thank you. So now we go back to the presentation, Tarun.
Very encouraging address to the audience. So without wasting much time, let us begin with the presentations. Good morning, good afternoon, and good evening to all there from various parts of the world. I welcome you all to this second day of DRPI 2023. And today we would have total 4 presentations from early stage researchers. So we did have 5 entries, but one entry was dropped. So the first entry would not be there from Australia. Please take a note of this, those who referred to the old agenda. All right. So I would like to just, clarify to all of you here that we would have 10 minutes of video recording. It's a recorded video presentation by the presenter. Then it would follow by 5 minutes of question and answer, which would be live, okay, between judges and the presenter. And after 5 minutes, judges would be given 5 minutes for evaluation and recording the remarks on the online system that we have developed for DRPI. So let's begin with our first presentation today. This is the presentation from DRPI, Japan and Southeast Asia. The title, from a biodegradable chemoradioimmobilization agent loaded with SM-153 radionuclide and doxorubicin. This is presented by Asil Arajib from Tyler's University, Malaysia.
Good day. Today my presentation is drug release from a biodegradable chemoradioimmobilization agent loaded with SM-153 radionuclide and doxorubicin. This will be the flow of my presentation today. Starting off, is liver cancer is the 6th most common cancer and the 3rd leading in cancer death worldwide. It's predicted that the incidence rate and the mortality rate to be increased by more than 55% by 2040. And liver cancer is the death by liver cancer is the 5th among males and 7th among females. The treatment options of liver cancer is based on the staging. So very early stage and early stage has more options of treatment such as ablation, resection, and transplant. While intermediate stages is transarterial embolization, advanced systematic therapy, and then terminal stages transplant or best supportive care. My focus will be about transarterial embolization. Now, what is embolization? It's when a procedure using particles or microspheres to block the blood supply towards the tumor, starving the tumor from all the nutrients and blood. In the case starting off, we have treatment, we have conventional TACE, which is conventional transarterial chemoembolization, where we have lipidol, the drugs, and embolizing agent injected towards the tumor, where the embolizing agent blocks the blood supply and the lipidol and the drugs go towards the tumor so that tumor cells can uptake the chemotherapy drugs. Next is DB-TACE, which is drug-eluting bead transarterial chemoembolization. The beads are loaded with the chemotherapeutic drugs injected towards the tumor, block the blood supply, and the release of the drug out of the beads. Next is TARE, which is transarterial radioembolization, similar to TACE. However, here we have a radioactive radionuclide loaded into the microsphere, injected towards the tumor, allowing the radiation to kill the tumor cells. So transarterial administering of the microspheres using the hepatic artery and injecting the microspheres towards the tumor in the liver to block the blood supply and also release the chemotherapy drugs or allow the radiation to kill the tumor cells. The uniqueness of this treatment is due to the liver itself, which has 75% of the blood supply to liver cells comes from the portal vein, while almost all the blood supply to the hepatic artery goes to the liver tumor cells. Therefore, when we're blocking the blood supply towards the tumor, we're not affecting all of the organ. Now, currently, TARE and TACE combined to get treatment is conducted by TARE followed by TACE, which is showed that median overall survival in the combined therapy is 36.8 months compared to TARE alone. However, there's limitation where there's no combination in one formulation or delivery system. Multiple treatment courses is required, therefore it's very costly and causes psychological burden on the patients. and has a higher risk of complications due to be admitted into the hospital multiple times. Another limitation is deuterium-90 is a radioactive or the radioactive radionuclide that's being used in transarterial radioembolization, which has no imaging properties, has a half-life of 64 hours. It's only produced in Singapore, Canada, and China on the whole entire globe. Therefore, it's very costly for other countries to request the deuterium-90 because it's a radioactive material. Therefore, the transportation is very costly and the production as well. So alternatively, samarium-153 will be one of the choices. It's a theranostic radionuclide, has a half-life of 46.3 hours, and can be produced by 223 reactors in 53 countries. Therefore, it's relatively cheaper to produce. As well, there is a lot of countries are possible to make the samarium radioactive. Therefore, they don't need to buy it from somewhere else radioactive. Therefore, cut down the costs in transporting radioactive material. Now, what's a theranostic radionuclide? Similar to samarium, it's combining diagnosis and therapy. So you have the gamma and beta emitters such as samarium, where the beta energy for the treatment of the tumors and the gamma for post-therapy imaging to ensure that the microspheres or the treatment has reached the correct or targeted location. The methodology of forming my formulation is using water and oil water solvent evaporation method, where we dissolved samarium, PHPV, and doxorubicin in chloroform. After that, the microspheres has formed, collected, dried in the oven, and then sent for nutrient activation. Now, nutrient activation occurred at the reactor at the Malaysian Nuclear Agency, where the samarium-153 was activated. So the nutrient activation with irradiation of 5 hours to convert the non-radioactive samarium into a radioactive samarium-153. After irradiation and cooling, gamma spectroscopy was conducted to ensure that there's no radionuclide impurities. So in the results, radioactivity of the DUX-SN-PHV microspheres had a specific activity of 8.68 gigabecquerel per gram, where the activity per microsphere was 177.69 becquerel per microsphere, compared to the commercially present deuterium-90 resin and glass, which had the activity per microsphere from 50 to 2,500 becquerel per microsphere. Therefore, this is an appropriate range for the current treatments, as well as compared with other papers which use samarium particles or microsphere. Activity per microsphere of DUX and PHPV was higher compared to other papers. Now, an analysis, a HPLC analysis was conducted for DUX before and after nutrient activation to ensure that the nutrient activation does not affect the DUX, and we had a similar retention time before and after. Now, using SCM, DUX-SN-PHV microsphere before and after nutrient activation, it kept its shape, which was spherical and no fragmentation. And the mean diameter was 13 micron with a size distribution from 25 to 37 microns, which is a suitable size for transarterial radioembolization to ensure that the microspheres are not too small, that they'll go to other organs such as the lungs, yet not too big that it will actually block the blood flow at an earlier stages far from the tumor. Now, the EDX also showed that there was no elemental contamination. So we had the samarium from the samarium, oxygen and carbon from the PHBV, and platinum from the coating for imaging purposes. Now, based on the thermal stability, the microspheres had to be able to withstand the temperature of the reactor, which will not top up more than 40 degrees Celsius. The thermal stability of the microsphere of DUX-SN-PHV was 155 degrees Celsius. Based on the FTR to ensure that the samarium and the doxorubicin were integrated into the PHBV, as well as the DUX-SN-PHV before and after nutrient activation does not have any elemental changes. So the peaks of the PHBV, the samarium, and the doxorubicin can be seen in the DUX-SN-PHV microsphere. Therefore, they successfully integrated. And the peaks for DUX-SN-PHV before and after nutrient activation was similar. Therefore, there was no elemental changes. Now, the radionuclide retention efficiency is a test to ensure that the radionuclide does not seep out of the microsphere, therefore will not go throughout the blood system and affect other organs. So it's tested in pH 7.4, pH 5.5, and blood plasma. So you can see the retention efficiency was very good, where it was more than 99%. This ensured even within 600 hours, which shows that the retention efficiency is quite high. Comparison with other samarium and other radionuclides, which usually is above to 95 or 98. Now, in vitro DUX release was conducted at PBS at pH 7.4 and pH 5.5. So at 7.4, 65.21% of DUX was released, and 29.96 at pH 5.5 up to 1,000 hours. This is to ensure that this is a desirable property of slow, steady release of the doxorubicin, so that even after the decay of the radionuclide, there's still treatment being released towards the tumor. So that even after the tumor cells that are trying to recover from the radiation, there's a doxorubicin which ensures that the tumor cells could actually die. And now the in vitro cytotoxicity test was conducted using MTT assay on HepG2 cell line, which is a liver cancer cell line. For comparison purposes, we used radioactive microspheres alone, the combined therapy of DUX and samarium, which is chemotherapy and radiation together, and chemotherapy alone DUX pH3 microspheres. So as the concentration increases, the cell viability decreases, and it's apparent that you can see the lowest cell viability was for the combined DUX and pH3 microsphere, and at 300 microgram and between 250 and 300 microgram of microsphere per ml. This shows that the combined therapy had more effect on the cell viability of HepG2 cell lines in comparison of chemo alone and radiation alone. The content of doxorubicin in the microspheres was 0.036 milligram per gram, and the samarium content was 12.8%. Current applications for the combined therapy, as you can see, other clinical trials have tried to conducted using systematic chemotherapy in deuterium microsphere, as well as TARE, transarterial radioembolization with sorafenib, and showed that the combined therapy had better results and compared to single. Now, for the potential applications of DUX SMPHPV microspheres is where we have the gamma, beta, and doxorubicin, where the doxorubicin and the beta will be for the treatment of the tumors, and the gamma is for post-therapy imaging to ensure that the microspheres reach the correct location, as well as it's more focused and concentrated of the radiation in one location. So in conclusion, a biodegradable chemoradiembolic microsphere
Tarun, the sound
DUX
is
and
gone.
SM153 was successfully developed for synergetic chemoradiembolization effect on liver cancer HepG2 cell line. Thank you very much.
Thank you, Asil, for that wonderful presentation. Krishna Priya ma'am, I believe everybody could hear the sound. Was there any problem?
Yes, just that, in the last little bit, there was a gap and then it became all right.
Okay.
I just want to request the judges whether you were able to hear.
Yes, I could hear it. Yeah,
I could hear it.
So we go on to the
I believe-
question and answer session.
Okay. Yeah. So we have our presenter here, Asil. Over to judges for questions and answers. Hello.
Who is starting?
So I'm starting. Good afternoon, good evening. A very nice presentation, very interesting topic. I will. We are several judges, so I will go quickly on two questions. You tested your microspheres for in vitro for drug release, and you tested them in two conditions: pH 4.5 and 7.4. So I have two questions. What is your rationale for selecting these two conditions, these two pH values? And the second question is, we observed a big difference in drug release between the two pH conditions. Do you have any explanation for such a difference?
Thank you so much for your question, and thanks for this opportunity. So starting off, the reason we used two different pH is because liver cancer in microenvironments, the pH is slightly acidic. So you want to compare the drug release, would it be still the same at the different pH, since while we're preparing this, everything is going to be at 7.4, but near the tumor location will be 5.5. So we want to ensure to see the drug release. Now for the second question, yes, the pH 5.5, the drug release is significantly lower compared to 7.4, which is kind of a desirable characteristics because we want to make sure that the release is slow enough so it will not become systematic, and we can still have treatment after the radiation is completely gone. So side note, the radiation will be completely decayed within 20 to 25 days. But at the same time, it's very, 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 very low compared, but this could be a possibility is due to that my pH3 microspheres are interacting with the ACAC of the samarium radioactive, Therefore, it's making a composite at the pores, making it quite difficult at pH 5.5, making it quite difficult for the ducts to be actually released, which will be furthering studying to see if this interaction is actually something that we can control, or is it something that we have to deal with or compromise for moving forward.
Excellent. Thank you very much. Good luck.
Thank you.
Maybe I can go next. Thanks a lot for the presentation. One question, maybe if you can explain, how would the two drugs that you chose would be distributed in the microparticles, and what is the mechanism through which the release is happening?
All right, so my samarium is the radioactive material. So it's a radionuclide which will not be released out of the particles, because then it will be actually be moving forward and moving everywhere in the body. But the radiation is strong enough or can penetrate enough to actually kill the tumor cells. So that's our target of the samarium. As for the doxorubicin, it will be released as any release of any similar to DB-TACE or TACE to be released towards the tumor. Due to we're using transarterial injection, so the microspheres will be injected towards the tumor near the capillary. So the diffusion and the radiation will actually affect the tumors at a very close proximity. And the other question, can you repeat it, please?
What will be the mechanism of the drug release from the microparticles? Can you give some insight there?
Because the microsphere has pores inside. So whenever, let's say the blood, or in the case of releases, my PBS is seeps into the pores, it will allow for the doxorubicin to detach from the PHBV and moving to the PBS or the blood towards the tumor.
Okay, right. Thank you.
Yes, Dr. Sandeep.
Thank you, Asil. Good presentation. Can you hear me?
Yes, I can hear you. Thank you.
Okay. Regarding the microsphere preparation, since they are prepared using, I think solvent evaporation method, I think that is what I saw, right? Number one is, how feasible that is commercially, and number two, how these microspheres would be sterilized, because they need to be administered parenterally, right?
Mm-hmm.
Okay.
All right, to be for the first question, we're using water and oil water solvent evaporation method. So when we want to upscale it, actually we can use a GMP lab to be able to use, for example, something similar to a bioreactor to do for the spindle and all the to double the amount, which is something we're trying to do, we're trying to do mass production, but it takes a lot of calculations of optimization. As for sterilization that we want to do is, since we be using a GMP lab and also using UV to ensure that all the microspheres, even after production and radiation, because we sent it to the reactor to be neutron activated to become reactor at the nuclear agency. So even after receiving it, we need to use UV to sterilize all the microspheres.
Yeah, just a follow-up question to that. Will UV be effective sterilization method? Because I'm not sure if it only be surface sterilization or.
Okay, because first off is that we are sending our microspheres inside containers, which will be using these containers directly towards the patient when we mix it. So getting it out of the reactor, it's already radioactive. So the radiation is quite strong already inside the reactor. So anything living thing will be dead by the time it gets out of the reactor itself. So then when we receive it, is just to ensure that all the outside containers, everything is as sterile as possible before using it. But then once the microspheres inside the containers are completely sterile.
Thank you.
Hello, wonderful presentation, lucidly presented, excellent work. I have only two questions because other judges have asked somewhat similar questions. There is one, what was the yield of this particular formulation? How much we started with and how much was produced eventually? The radionuclide yield.
The yield, it was we started off with PHBV microsphere, which is 0.1 gram. And this is with one run. We ended up with around approximately 300 milligram of microspheres at the end of the production.
300 milligrams from, sorry, I couldn't hear you first.
I'm sorry, can you repeat?
Can you please repeat?
Based on yield, we started with 0.1 gram of
100 milligrams.
100 milligram, yes.
Okay.
Oh no, sorry. I was
Yeah, that's a bit.
yeah, sorry. And ended up wait. Right. Oh yeah, it was 0.15 grams, so around 150 milligram. Yeah, and we ended up with 120 milligram.
Good.
But we're trying to optimize because during filtration, the filtration method we use in the lab was quite.
So what was the filtration method? That's what I'm coming to actually.
The filtration method we use centrifugation because we couldn't use a filtration pump because the doxorubicin microspheres will get stuck into the filtration kit.
Yeah.
So we had to use centrifugation.
So what was the optimized G value of the centrifugation?
Again, can you repeat the question?
What is the optimized centrifugation rate?
We ate it from 8,000 to 8,500 RPM for 10 minutes. This is for collection, and then for washing, we also did the same thing, but for 6 minutes.
Will that affect the yield? Any change in centrifugation?
It affects us slightly, but we needed to use that kind because the microspheres are quite large, so they are supposed to settle faster. But we still needed to centrifugation to be enough so that any loose particles of samarium or doxorubicin actually can be floating. So if you go higher, you'll actually have a strange yield of other particles present.
So just to continue on that question, I wanted to know how was the safety measures? Was it done in the laboratory, in your laboratory? And second thing that I will continue to ask is, what was the storage of this particular compound? I mean, formulation. How did you store it?
Can you just repeat the first question? I couldn't hear.
What are the safety measures in your laboratory?
Yeah. So the uniqueness of using samarium is that I can do all the preparation in normal pharmaceutical lab because there's no radiation. It will only become radioactive once we send it to the nuclear reactor to be activated. Then from there, we can actually send it to the patient. So there is not needed except the general PP for the lab. So because we're using chloroform and the fume hood. But after it's been radioactive, it gets sent over and it's quite, so that's one of the benefits compared to its commercial, which is Yttrium-90. If we need to use the radioactivity, everything has to be radioactive. So we need to be using lead and all the protective measures. As for storage of the microspheres, so after the microspheres have been dried, they'll be stored at 4 degrees Celsius in a fridge.
Thank you. Well presented. All the best. Good work.
Thank you.
Hello, Asil, can you hear me?
Yes, I can hear you, Doctor.
Yes, hello. Well, thank you very much for a very, very interesting presentation. Just one small question. When you had irradiated it, you used HPLC to confirm that it hadn't degraded. You only mentioned the retention time though. So what I would probably expect is if it had degraded, it might only be a small amount. So the main peak would still be there at the same retention time, but there would be other ones. So the two things I wanted to ask is, did you look at peak area to indicate whether it was still, whether it had degraded at all? And also, did you look for the presence of any degradation products in your HPLC, rather than just relying on the retention time?
Thank you. We looked up for over around 25 minutes of the HPLC to see if there's any peaks have been present, but we couldn't find except the singular peak. The goal is that for the future that we want to use an LCMS so that we can have a more accuracy. However, unfortunately, we did not have any currently, and our neighbors also did not have. So that's one of the targets in the future because as we know that radiation has effect. However, based on other papers where they irradiated doxorubicin at different irradiation for a long period of time, there was no degradation present. It was only degradation was observed when using acidic condition and very high temperatures. So we based our study also on those studies until we can actually do more investigation for the doxorubicin degradation products.
That's great. Thank you very much.
There are no more questions, then we can The judges can put their marks and their remarks, and Tarun can play the ad.
Thank you very much.
And thank you.
Thank you.
All the judges have finished with their evaluation, then we can proceed for the next presentation. Tarun?
We can start the next presentation, Tarun.
Tarun?
All right, so here we go with the second presentation today. The title is Effect of Surfactant on the Drug Release Behavior for Amorphous Solid Dispersions. This is coming from DRPI India, and the presenter is Prachi Srivastav from National Institute of Pharmaceutical Education and Research, Sasnagar, Punjab.
Hello, I will be speaking on the topic effect of surfactant on the drug release behavior from amorphous solid dispersions. Polymeric amorphous solid dispersions are formulated to address bioavailability challenges of poorly water-soluble drugs. The dissolution enhancement from ASDs is higher than the neat crystalline drug and the neat amorphous drug at low drug loadings, beyond which drug release tends to fall. Additives such as surfactants are being investigated to improve drug release without compromising drug loading. However, surfactants can also negatively affect ASD release due to induced crystallization. Hence, careful selection of surfactant is essential to achieve an optimal release performance. For the model ASD system, the API chosen was efavirenz, which is a BCS class 2 drug and a fast crystallizer. After screening several polymers, HPMC-AS-LF was selected to form ASG with efavirenz. The ternary component was a widely used anionic surfactant, sodium rhodosyl sulfate. Binary and ternary ASGs were generated using spray drying at 6 drug loadings from 5% to 50% weight ratio. SGS was added as 5% ratio of total binary ASG weight, such that its concentration remains below CMC in dissolution setup. After formation, the ASGs were characterized for their solid-state characteristics. This is a morphology under scanning electron microscope. In polarized light microscope, the absence of birefringence indicates the amorphous nature of the ASG particles. Under X-ray powder diffraction, the absence of characteristic diffraction peaks indicate the conversion of amorphous form. In infrared spectroscopy, the absence of the OH stretching of HPMC-AS, NH stretching of efavirenz, and the reduced intensity of the carbonyl stretch of efavirenz indicates the formation of intermolecular interactions. In differential scanning calorimetry, the absence of melting endotherm in the ASGs and the presence of glass transition temperatures indicate the conversion to glassy state. The TG is reduced with increasing drug loading. In case of ternary ASGs, the TGs were reduced by around 6 degrees Celsius due to plasticization. Powder dissolution was conducted in PS 6.8 phosphate buffer under non-stain conditions. This is a release profile of binary ASGs. This horizontal line depicts the amorphous solubility, which represents a maximum achievable free drug concentration. Drug loadings up to 20% weight ratio release drug beyond amorphous solubility, whereas higher drug loadings show poor release. The Resigno index obtained by comparing drug and polymer release showed a drastic difference between 20 and 30% drug loading. This is a release profile of ternary ASGs, where the drug loading dependent drastic difference was absent, but during dissolution, precipitation had occurred for all ASGs, which shows the dissolution advantage was realized. Hence, the Resigno index did not show any abrupt change within drug loadings, and the solution concentration achieved was higher in ternary ASGs as compared to binary one, especially for the 3 highest drug loadings. For the mechanistic understanding, first the bridge between the molecular level property, that is the diffraction content, and the particle level property, that is the morphology under cell, was correlated by dissolution by precipitation studies. The needle-like precipitates were correlated to DFT crystalline efavirenz. Solid-state events were thus monitored using real-time optical microscopy, and the samples were collected at 2 different time points to observe the occurrence of recrystallization during dissolution. It is more evident under real-time scanning electron microscopy, where matrix crystallization was observed from 30% drug-loaded binary ASGs, and for ternary ASGs, the crystallization was observed for samples collected after precipitation from 5% drug loading upwards. This also gives an indication that in ternary ASGs, there may be heterogeneous nucleation on these crystallized ASG particles, which may lead to bulk precipitation. The liquid state events were monitored for the size distribution of drug-rich nanodroplets, which are formed when the amorphous solubility is exceeded due to LLPS. In binary ASGs, up to 20% drug-loaded ASGs could form these nanodroplets within a size range of 150 to 400 nanometers, whereas in ternary ASGs, even 30% drug loading could form the nanodroplets. But unlike binary ASGs, these nanodroplets did not sustain up to 120 minutes of dissolution due to precipitation, as mentioned earlier. To probe whether water-induced amorphous phase separation was the precursor to poor release, the phase behavior studies was conducted and films were observed under CLSM. So, for the films stored at 97% relative humidity, as well as for films immersed in buffer, the resulting films did not show any localized migration of dyes, which means that ALPS did not occur in them. These results were also supported by DSC, where these spray-dried ASGs were stored at 97% relative humidity for 48 hours, and no separate drug and polymer DGs were observed, which means APS was not the precursor to matrix crystallization. The self-association of drug molecules in dissolved state can stabilize the supersaturated state. In ITC experiments, the enthalpy required for forming self-associated structures of efavirenz was lower in presence of HPMC-AS alone than in the presence of HPMC-AS and SGS, which gives another reason why this supersaturation was not stabilized in ternary ASGs. This slide shows the summary of the dissolution mechanism of binary ASGs. For the low drug loadings, the drug release was good, such that there was nanodroplet formation after the bulk drug concentration exceeds amorphous solubility, and there was absence of matrix crystallization up to 120 minutes. Whereas for the higher drug loadings, poor release was as a result of the crystallization from the ASG matrix. In case of ternary ASGs, if we look at the low drug loadings, so due to faster wetting kinetics by SGS, there was comparatively higher drug release. But at some point during dissolution, there was initiation of matrix crystallization. And not only that, but these matrix crystallized particles acted as seeds for primary heterogeneous nucleation, which led to the bulk drug precipitation. And the drug-rich nanodroplets are depleted to compensate for the lost drug. For the intermediate drug loading, that is 30%, there was good drug release and nanodroplet formation, which wasn't observed in binary ASGs, which is an advantage. But the subsequently, the crystallization and precipitation had occurred in these as well. For the high drug loadings, even though an initial higher drug release as compared to binary was observed, but the overall release was still poor due to crystallization. This slide shows the morphological correlation with drug release without surfactant, and this was with surfactant showing the biphasic release pattern. In conclusion, due to the plasticization ability, SGS triggered matrix crystallization even after an initial high drug release. Nanodroplets formed at around 10% drug loading higher due to faster wetting kinetics by SGS. Ternary ASG particles induced bulk precipitation through heterogeneous nucleation. This suggests that surfactant choice is very critical to avoid failure of ASGs through crystallization while also maintaining the above mentioned benefits on drug release behavior. Thank you very much.
Thank you, Prachi, for that nice presentation. Over to judges for Q&A. Prachi, you may please start your camera and mic.
Yes, Dr. Sandeep Tiwari.
Yeah, thank you, Prachi. Good presentation. I just have two questions actually. Number one, your choice of surfactant was based on literature survey, I believe. You selected only SDS and not others, and if that is true, I just would like to hear. Number two, what was your limit of congruency for the drug polymer combinations, because it seems you are simply using a surfactant to ensure that you are able to get parachute effect with the use of a surfactant. So did you determine the limit of congruency for your drug polymer combination? Am I audible? Sorry.
Hear you.
You cannot hear?
No, we cannot hear Prachi.
Okay.
Hello. Hello, am I
yes,
audible?
yes. Now you're audible.
To answer your first question, yes, we selected SDS based on literature because it was the most widely used surfactant and marketed ternary ASG formulations. And could you repeat the second question because at that time I could not hear?
Yeah, the second question is related to limit of congruency related to your drug to polymer combination. Did you determine that? Because I see that you selected drug loadings from 5% to 50%. So
Yeah.
did you find your limit of congruency for this combination, for the specific drug to polymer combination?
Yeah, yes, sir. So we selected these 6 drug loadings precisely to see the limit of congruency of our binary ASGs and whether we can improve that using ternary ASGs. So our limit of congruency for binary ASGs was 20% drug loading, because about 20% drug loading, when we increased it to 30% drug loading, these ASGs did not exceed amorphous solubility and showed poor release. So that was our rationale.
Thank you.
Rashi, thank you for your presentation. Very briefly, but based on experience, we know that having a fully characterized surfactant is complicated, and there could be variation from supplier to supplier or even from batch to batch. Is it something that you explored to evaluate if you change the supplier of SLS, if you are obtaining the same outcome, or is it still something on your way?
Yes, so that's a good question. Yes, especially it is true for SGS where there are chances of residual content remaining. But we did not check the different suppliers in this study, but we did use SGS from one single supplier for all the studies in this experiment. But it remains to be seen in the future.
Okay, but that could be something to explore further, because for dissolution testing, it has been demonstrated already that from one supplier to the other and so on, the effect on dissolution behavior could be different, and I would expect to have to see some differences on your product, on your formulations. Okay.
Yes, it remains to be seen. We will explore it further.
Thank you. Good luck.
Thank you.
Prachi, just a follow-up question here. Besides surfactant, did you evaluate any other polymeric crystallization inhibitors to see if they are better than surfactants?
Yeah, so it's true that ternary ASGs could also be formulated using a second polymeric component. But in this case, we did not use another polymer. The reason we used surfactant was to reduce the pill burden. So if we use another polymer, it will only add to the weight. And since if many drugs, such as efavirenz itself, has a dose of 600 mg, so we try to use surfactant to improve the drug release.
Thank you.
Are there any more questions?
Just one more from me.
Yes.
Okay. Hi, Prachi. Thanks a lot for the presentation. Did you conduct any stability studies on these ASDs, both the binary and the ternary? And what was your, if yes, what was your outcome?
Thank you for the question. Yes, we did stability studies for 30 days. This was to confirm the amorphous nature and confirm the amorphous nature of the ASGs before dissolution experiment. So we did stability for that purpose. But long-term stability, accelerated stability studies is yet to be done.
Okay. Thank you.
Okay, Dr. Srinivas Patnala.
Thanks for your good presentation, actually, and surely there is a lot of prospective outcomes of this research. Only one question I have, because others have already asked that question, some of the questions. Only thing that I wanted to know is, in your abstract, you have mentioned that non-sync powder dissolution. It says that EFV and HPMCAS concentrations were determined using HPLC and colorimetry, respectively. So what was the need to do such analysis?
Yes, so the drug content was determined using HPLC and the polymer content was determined using colorimetry. There is a colorimetric method for HPMCS determination, which is much easier than the size exclusion chromatography, which is usually done for polymers. So we used HPLC for drug and colorimetry for polymer.
Okay, thank you.
Are there any more questions from the judges? Okay, if not, they can Mark and Tarun, you can start with the ad.
Thank you
you,
very much.
Rajiv.
Thank you.
Have the judges finished with the evaluation?
It's done for me.
Okay, Tarun, we can proceed.
Yeah, so we start with the third presentation in the series, which is basically 04 number on the evaluation sheet. This is coming from Bharti, from National Institute of Pharmaceutical Education and Research, Ahmedabad, India. The title is Formulation Development and In Vitro Characterization of Hot Melt Extruded Cyclosporin Ocular Inserts for Dry Eye Disease.
Warm greetings to one and all watching this video. I'm here to present my research work entitled as Formulation Development and In Vitro Characterization of Cyclosporin Chloride Ocular Inserts for Dry Eye Disease. So dry eye disease is a multifactorial condition that occurs due to any dysfunction in the glands that are responsible for the formation of the tear film. The tear film is basically composed of 3 layers, namely the lipid layer, the aqueous layer, and the mucin layer, and any dysfunction in one of the 3 layers would result in an increase in osmolarity of the tear film. This eventually leads to a cascade of immune responses such as epithelial cell membrane disruption, damage to the goblet cells, alteration in the protein-lipid interactions of the tear film, and triggers a release of inflammatory cytokines, causing ocular irritation and inflammation. Cyclosporin is well known for its anti-inflammatory effects, and it has been intended for the management of dry eye disease. However, the currently available marketed formulations are in the form of nanomicellar solutions and emulsions, and these are associated with numerous limitations such as low ocular viability and a high frequency of administration. Here, we intend to develop inserts to reduce the frequency of administration and enhance patient compliance. The drug and the polymer are fed into a hot melt extruder, and the resulting extrudes are then fed into a stretching and cutting machine called a calicut that gives us the inserts of desired dimensions. Optimization of process parameters of both HME and the stretching and cutting machine were carried out. Initially, we studied the effect of pre-mixing step on the content uniformity. As we all are aware that a large quantity of drug usually goes into HME, as in the case of amorphous solid dispersions, where around 15 to 40% of the drug is loaded in a polymeric matrix. However, for ophthalmic indications, the dose required is generally low, and here we have incorporated only 1% of the drug. So achieving content uniformity was a major challenge. We compared 3 different pre-mixing steps, namely geometric mixing, wet mixing, and we found that wet mixing yielded inserts of good content uniformity. Then we also optimized the barrel temperature and the screw speed. Here we used the one factor at a time approach, where initially the screw speed was kept constant and the temperature was altered to identify the optimum temperature for good extrudability of the inserts. Then we kept the temperature constant and the screw speed was altered. So the table here depicts the optimum temperature for good extrudability molecular weights of the polymer hydroxypropyl cellulose. We also studied the effect of the screw speed on in vitro release, and we found that there was no significant difference in the release profile. So this in turn establishes the robustness of the mixing that occurs inside the barrel of HME. Here we also study the effect of the heater temperature of the stretching and cutting machine on the sharpness of edges. So basically, though HPC is approved by the FDA for ocular indications and it is biocompatible, here we can define biocompatibility not based on the material property but based on the surface characteristics of the dosage form. So here we could see that when the operation was carried out at 25 degrees Celsius, the edges of the inserts were sharp. However, when the same unit operation was carried out at higher temperatures, say 45 and 55 degrees Celsius, inserts of relatively smooth edges were obtained, which would be more compatible than the previous ones. Then, after fabrication of the inserts, evaluation was carried out. The weight of the inserts were around 5 mg and the average dimensions were 3.3 into 1.27 mm. The surface pH of the developed inserts were 7.3, that was in accordance with the ocular pH. The drug excipient compatibility was carried out by ATR spectroscopy, and here we could see that the characteristic peaks of both cyclosporin as well as HPC is retained in the ATR spectra of the formulation. So this indicates that there were no significant interactions between the drug as well as the polymer, and that is why the peaks have been retained in the ATR spectra. The inserts were also characterized for swelling index because swelling is an adhesiveness of the inserts once it is placed inside the cul-de-sac. The results of scanning electron microscopy also revealed the smooth surface of the inserts. So since there is no compendial method available for the dissolution testing of ocular inserts, we developed a novel in-house in vitro dissolution method. Here we placed a stainless steel mesh in between the magnetic bead and the insert in order to prevent accelerated dissolution of the inserts. In the absence of a stainless steel mesh, what we observed was that the magnetic bead would often interfere with the dissolution profile of the inserts, giving us a false profile. However, when we developed the mesh with certain perforations and spaces, we were able to find that efficient circulation of the media was obtained, and this method was also able to discriminate the dissolution profile of various molecular weights of the polymer, hence enabling us to achieve the identification of product to product variability. So for the selection of dissolution media, initially we carried out equilibrium solubility studies, as we know that there is a criteria that the dissolution media should be able to dissolve 3 times higher the quantity of drug that is present in the formulation in order to maintain sink conditions. We selected 0.25% SLS as our final media as it exhibited the highest solubility. Based on the release profile that was obtained in both 0.1% SLS in PBS versus 0.25% SLS, we could see that 0.25% SLS gave a better release profile, and therefore we have selected that dissolution media. Further, the volume of the dissolution media also is critical here, because only 50 microgram of the drug was loaded into the formulation, and using a higher volume of the dissolution media would be challenging for detection through HPLC, and hence we restricted to only 5 ml of PBF. This is the in vitro release profile of the inserts, and here we could see that the MXF grade of HPC, that is around 850,000 Daltons, was able to give a sustained release of the drug for a period of 24 hours. And also the developed dissolution method was able to differentiate between the release profiles of various other molecular weights, thus proving the discriminatory power of the dissolution method that has been developed. Here, the in vitro degradation of the inserts are also in accordance with the release results. Here we could see that the MXF grade was degraded at 24 hours, where only less than 10% of the polymer was remaining. So this is in accordance with the in vitro release profile, that the MXF grade is ideal for fabricating inserts that are intended for once in a day administration. The inserts were also characterized for stability over a period of 3 months, and here we could see that there was no significant change in the percent drug content after 3 months of storage at both room temperature as well as accelerated conditions. The not significantly different, they were established. Vivo eye irritation studies were carried out where one eye of the rabbit was left untreated, that served as a control, and the other eye was the treatment. And based on race scoring system, we could see that there were no significant differences in both control and treatment, and hence score. So I'd like to conclude by saying that the developed cyclosporin-loaded oculi inserts were able to give a sustained release of over 24 hours and were found to be stable at 3 months of storage. And the in vivo biocompatibility studies also exhibited no eye irritation. So the developed formulation could be used as an alternative to marketed formulations, as it has a low frequency of administration, that is just once in a day, as compared to the marketed ones, which needs to be administered 4 to 5 times a day for management of dry eye disease. Thank you.
Thank you, Bharti, for that nice presentation. Over to judges for question and answers.
Yes. Thank you, Bharti, for good presentation. I have one question. You mentioned that you use HPC, right? And I think you evaluated different grades. Can you tell us what is the TG and the degradation temperature for HPC, number one? And the second question is, was actually, I just lost my thought process, but I think I would like to hear your answer for the degradation temperature and TG, and if you used any plasticizer in this, because I did not see it in your presentation for extrusion.
Yeah, so the TG of HPC MXF grade is around 105 degrees Celsius. So in HME, we usually process 30 degree above the TG. So the processing temperature was around 140 to 150, and the degradation of HPC is above 200. So it was ideal to operate at this temperature. And we did not use any plasticizer because plasticizers are generally used when the TG of the polymer is way higher, around 180, and we need to reduce the TG. But here, since the TG is already low, we did not have a need to use the plasticizer.
Thank you.
Can I go next?
Yes.
Yeah, thanks, Bharti, for the presentation. Very interesting work. Did you or do you have any insight on what is, is there a need to sterilize such implants for ocular delivery? And if yes, what impact would sterilization have on HPC extrudates?
Yeah, so according to me, the ideal method of sterilization would be gamma irradiation. And I think there would be no effect because HPC is a non-ionic polymer. And so the ionizing effects of gamma would not have an impact on the polymer and the drug as well, I guess.
Did you evaluate it experimentally?
No, we did not have access to the gamma irradiation facilities. Yeah.
Thank you.
Who's going
Hello,
next?
hello, Bharathi. Very interesting presentation. So I'd just like to ask, first of all, if you could explain how this treatment is administered, and secondly, did you do any toxicity studies other than the irritation studies that you showed us?
No, we are currently working on doing the efficacy and toxicity studies, but with regard to the administration, the inserts would be placed in the inferior cul-de-sac of the eye. So we need not do any surgical procedures that is required with other ocular inserts such as punctal plugs, or so it is really patient friendly. We can just insert it with a administrator, self-administrable, or just as we insert contact lenses.
Thank you.
Good presentation, good work there. I think my previous judge already asked one question about stability, which was also one of my questions. But continuing, do you make this formulation as a unit dosage form, right?
Yeah.
So what will be the kind of packaging for this?
So yeah, it would be aluminum packed, and I think we can pack each unit in a different pocket and then give it as a, say for example, a monthly 30 different packs for we can give it.
In terms of, just a curious question, in terms of if you pack it like that, then what is the way the person is going to insert? You just mentioned that it can be inserted in the cul-de-sac, which I agree. But how is it going to be handled? Because the stability is lost.
No, we could do terminal sterilization, and I think so it would be peelable. Like we can make a packaging container which has how do I say it? We just peel off the upper layer, and then we get access. Just how we pack ocular contact lenses. Yeah.
I don't know, but application, and probably it requires a lot of dexterity of the hand, and I don't know what.
I think we could design an applicator with pointed edges and just like a forceps or something using plastic, and we could reuse the applicator every day, I guess.
Probably that's okay. There's one section you have done the dissolution studies at 34 degrees centigrade. Any particular reason?
Yeah, we refer to the literature, and it is reported that the ocular temperature is quite lower than 37. So in order to mimic that, we used 34.
Good. Thank you.
Yeah.
And just a quick one to finish this session. Thank you for your presentation. You show drug release profiles increasing, and also a graph showing the weight loss of the implant. Do you think that this, and you mentioned that the one was correlating the other, do you think that the weight loss is accurate enough, knowing that HPC may be subject to hydration, and there could be a loss of polymer, but could be compensated by hydration? Did you consider that?
Yeah, I think the more accurate technique would be size exclusion chromatography in order to study the degradation profile. But yeah, this was just to prove that the inserts, the HPC gets hydrated and gets dissolved within a period of 24 hours. So we just mimic the same setting as the dissolution in order to see is it really getting dissolved within 24 hours, and how the weight losses, because it's the mechanism would be swelling followed by erosion, right? So yeah, we just try to characterize that with a simple technique.
Very good. Thank you very much. Thank you.
Yeah. Thank you.
One last thing I was waiting.
Yeah.
This is more like a discussion rather than a question. That's why I was waiting for the panelists' opinion. Thanks for giving the time. One thing that I wanted, cyclosporine is also used for organ transplant. Have you looked at the possibility that it can be this kind of a formulation being used for organ transplant, so that, because that probably is more problem for most of the ophthalmologists. They use 5-FU solution to get that kind corneal transplant. So and also the rejection rate is quite high from what I understand. Have you looked at this?
I think we can design such a formulation, but the issue would be we would need a higher dose for such an indication, and that would in turn, I don't think HPC would be an ideal polymer because it gets dissolved within 1 day. So we would want to select a polymer that gives a sustained release for over 1 month or 3 months. So I think we need to consider the polymer again, and yeah.
Thank you. Thanks. Good luck.
Thank you.
Arun, you can play the ad. If everyone has finished with evaluation, we can go to the final presentation of today. Tarun?
All right, so let's move on to the final presentation for today. The title is Supersaturable Bioself Nano-Emulsifying Drug Delivery System for Improving Oral Bioavailability of Dasatinib. This presentation is coming from Naipur, Sasnagar, Punjab, and it is presented by Darshini.
Greetings to all. I'll be presenting the research project entitled Supersaturable Bio-Self Nano-Emulsifying Drug Delivery System for Improving Oral Bioavailability of Dasatinib. Dasatinib is a BCS class 2 drug having poor aqueous solubility and poor oral bioavailability when administered in the form of conventional oral dosage forms. So, we use SNITs as a formulation strategy to overcome the drawbacks associated with dasatinib delivery. Supersaturable biosnits consist of the drug dissolved in a combination of synthetic oil and a bioactive oil with aid of a surfactant and co-surfactant. Along with this, a precipitation inhibitor is also incorporated to improve the drug loading without causing precipitation. This anhydrous preconcentrates when they come in contact with the aqueous GI environment, and due to the peristaltic movement, they'll form emulsion globules inside the body. These emulsion globules will be subjected to digestion. The digestion products like monoglycerides, triglycerides, and fatty acids will form micellar and vesicular structure with the drug. These colloidal structures will act like a reservoir and they will enhance the solubility and permeability of drug. They also promote lymphatic transport. The main challenge over here is supersaturation. Since a drug has pure solubility in intestinal pH, it will be present in a supersaturated state and hence highly prone to precipitation. In order to prevent this precipitation, we are incorporating a precipitation inhibitor. These are the major objectives of the study. To start with the selection of excipients, we performed solubility studies in various excipients. Among the synthetic oils which were screened, dasatinib showed maximum solubility in oleic acid. And among the natural oils which were screened, dasatinib showed maximum solubility in chamomile oil. Hence, oleic acid and chamomile oil were selected as the oil components of our formulation system. Further, surfactants and co-surfactants were screened based upon their emulsification ability. For this purpose, emulsions were prepared with various surfactants, and the prepared emulsions were characterized for size, PDA, and transmittance. Based on these results, Chromophore RH40 was selected as the surfactant. A similar protocol was followed for screening of co-surfactants also, and transcutol HP was selected as the co-surfactant. After selection of excipients, we need to optimize the concentration ratio of each excipient. For this process, we used design of experiment approach. We used optimal mixture design and formulated 13 formulations containing 13 different ratios of all the excipients. These formulations were characterized for size, PDA, and transmittance, and based upon these responses, a final optimized formula was derived at the end of DOE trial. Moving to the screening of precipitation inhibitor, the normal in vitro supersaturation test do not take into consideration the digestion process. But in lipid-based formulation, digestion plays a crucial role in determining the amount of drug which will be available for absorption. Hence, we used an alternative approach for screening of precipitation inhibitors, that is the high throughput in vitro lipolysis method. In this method, we prepared formulation containing different precipitation inhibitors and we subjected them to digestion process. At regular intervals, samples were withdrawn and the samples were centrifuged. On centrifugation, we were able to get 3 distinct phases of undigested lipid phase, aqueous phase, and the pellet phase. Among these 3, the aqueous phase consists of drug which is present in the colloidal forms, and this is very crucial because only this aqueous phase concentration will be available for absorption. So we screen various precipitation inhibitors based on their ability to maintain high drug concentration in the aqueous phase for a longer period of time. We also calculated the supersaturation ratio. Based on these results, PVP-K30 was selected as the precipitation inhibitor. We prepared formulations with increasing concentrations of PVP-K30 and we subjected them to the HTP method. Based on the results, 2.5% PVP-K30 was finalized as the optimized precipitation inhibitor concentration. The final optimized formulation was characterized. It was found to have a nanometric droplet size and spherical emulsion globules. Further, the precipitates obtained from the lipolysis experiment was also characterized using SEM and PXRT. These analyses revealed that there was amorphization in the drug which was achieved using the formulation. The formulation was also found to be robust to dilutions and it was found to be stable in simulated gastric and intestinal fluids. Coming to the drug release studies, the conventional dissolution method or the dialysis bag method will not hold appropriate for lipid-based formulations. As I earlier mentioned, the process of digestion is very crucial in determining the in vivo fate of lipid-based formulations. This led to the emergence of several different digestion protocols and different techniques for evaluating the in vivo fate of lipid-based formulations. On this note, we used a simultaneous lipolysis permeation method to study the drug release pattern from our formulation. In this method, we dispersed our formulation in the lipolysis media and the digestion was initiated by adding pancreatin enzyme. On initiation of digestion, triglycerides, monoglycerides, and fatty acids will be produced. These fatty acids will cause reduction in the pH of the media, and this pH drop will be sensed by the pH electrode, and an equivalent quantity of NaOH will be added to maintain the pH to 6.4. The pH 6.4 has two significances. One is that activity of enzyme will be appropriate only at pH 6.4, and the other one is that the volume of NaOH consumed during the experiment will help us to predict the extent of digestion of our formulation. So at different time intervals, two sets of samples were withdrawn. One set was directly subjected to aqueous phase concentration determination, and the other set was transferred to the donor compartment of transdiffusion cell, and the permeation across dialysis membrane throughout the 3 hours was studied. The in vitro lipolysis results showed that our formulation was able to maintain 13.5-fold higher concentration in the aqueous phase compared to the suspension. The permeation profiles also revealed higher permeation achieved using the formulation. Further, we also calculated the steady-state flux and apparent permeability, and this also revealed that the formulation showed significantly higher permeability coefficient compared to the suspension. Further, in vivo pharmacokinetic studies were performed on SD rats by oral administration of 10 mg per kg dose. These studies revealed that formulation was able to achieve 2.7-fold and 1.8-fold higher AUC and Cmax compared to the suspension. Establishing IV-IVC for a lipid-based drug delivery system is a complicated challenge and it is still being debated. Even though many experiments and reports have explored the potential of pH-stat lipolysis method, still it lacks in establishing IV-IVC. Hence, in the current study, we combined the pH-stat lipolysis to a permeation setup and we explored its potential in establishing IV-IVC. For this purpose, we calculated the cumulative permeated amount in vitro and we compared it with the in vivo AUC. On plotting the in vitro and in vivo AUC of all 3 formulations, we were able to get a linear correlation with R-square 0.9042. To conclude, the developed supersaturable biosnits was found to be a potential drug delivery strategy to improve the solubility and bioavailability of dasatinib. And the explored simultaneous lipolysis permeation method was found to be a promising tool in estimating the in vivo performance of SNIDs. The future prospects of this project include application of mathematical models to establish better IVIVC, performing in vitro cell culture studies using CACO2 cell lines, evaluation of other model lipid-based formulations using this method in order to check the reproducibility of the method, and using the supersaturation strategy for other anti-cancer drugs as well. Thank you for your patient listening.
Thank you, Darshini. And over to judges for question and answers. Dr. Patnala.
Who is going to begin?
I can quickly start. Thank you, Darshini, for the presentation. I'm sorry, maybe I had a loss of connection. You mentioned that the regarding the characterization methodology that you are using with this lipase and so on, digestion, the pH, the selected pH was something like 6.4 or something like that. Okay. What? Yeah, and it's adjusted continuously in the first beaker.
Yes, sir.
Okay. How was that value selected, and what is the rationale behind that value?
Yes, sir. Actually, the lipolysis setup was mainly made to mimic the upper intestinal part, because the lipid digestion, a major portion of lipid digestion takes place in the upper part of the intestine, in the duodenum region. So we use the standardized protocols. Means like unlike the solution methods, there are no definite guidelines for these systems. But LFCS consortium, developed series of 6 papers standardizing each of these conditions. And according to that, in order to mimic the intestinal pH, we took the value of pH 6.4 to 6.5. And the pH is maintained so that the enzyme activity is not lost throughout the experiment.
Thank you
Yes.
for the clarification. Thank you.
Any more
Hello,
questions?
Darshini. I have a similar type of question related to the oils that you chose. Could you explain the reason why you chose those particular oils? And I noticed you said oleic acid was synthetic. Was that, were you referring perhaps to a different oil there? So could you just tell me a little bit about the oils that you chose at the start, please?
Actually, in case of oil selection, we used the solubility as a determining factor. We tried with various synthetic oils like medium chain and long chain triglycerides, capmol, capriol, and everything. But the solubility was like a good amount in only in oleic acid. So we chose that as the synthetic oil. But our initial aim was to use a bioactive oil. That is a natural oil which is having anti-cancer activity. So that's why we incorporated this natural oil part also. But that part, that oil is not sufficient to maintain the solubility of the drug. Like we can't achieve the desired drug load using that oil alone. So we have used a mixture of synthetic oil and natural oil. Yes, sir.
Thank you, and thanks for a nice presentation.
Thank you,
Dr. Sandeep Tiwari.
Yeah, thank you, Darshini, for good presentation. Taking the same question a little bit further from Dr. Stephen, with regards to your precipitation inhibitors, polymeric precipitation inhibitors, can you remind me which ones you selected for screening, and if you ever evaluated Soluplus or Colidone BA64 in your screening?
Yes, so we haven't tried Soluplus, but we evaluated PVP-VA also. We tried PVP, K30, HPMCAS, and PVP-VA, and also other few polymers like Udrage. And the main problem was initially we filtered the polymers based upon the solubility also. Means few polymers were not solubilizing in the formulation easily. So in the initial stage itself we filtered, and after that we proceeded with these polymers which I mentioned for the precipitation studies. And among that we found out that PVP K30 was able to maintain the supersaturation state for a longer period of time. So we went ahead with PVP K30. We tried PVP-VA also, sir, but it
it also showed good, good precipitation inhibition. But since we found this K30 to be better, we went ahead with that polymer, sir.
Okay, sure. Thank you.
Yes.
Do we have time for one more question?
Yes, yes.
Okay, thanks, Darshini, for the presentation. A question on, like, if this was to go forward into the clinic, what would be, or how would you present your formulation in a dosage form? What would be your preferred dosage form going forward?
Actually, we can go for either soft gelatin capsules. We can fill these pre-concentrate, liquid pre-concentrates which we have formulated into soft gelatin capsules. That would be a great option. Or in case if we want to improve the stability more, we can absorb it into like few adsorbents like Mucilin or Aerosil, and we can go for tablets or hard capsules also. So we have like these options to proceed further.
Yeah, thank you.
Thank you, sir.
Maybe one more. Okay, please go ahead. No, please go ahead.
No.
I was just curious, like you said, you evaluated a lot of oils, MCTs, long chain triglycerides as well, but you found good data with oleic acid. Do you have any hypothesis of why do you see a very good solubility on oleic acid?
Yes, actually, there are few reports like the oleic acid chains, the hydrophobic chains give more, they have kind of ionic interaction also with the dasatinib molecule. So there are like very few, reports regarding that. So that might be a possible reason why the drug was showing good solubility in oleic acid.
Okay, thank you.
Good presentation, good work, Darshini. Just one question. I think most of the questions are already asked, so I will not. So why did you choose dasatinib? Because I think the other one, the imatinib, is still requiring. Anyway, dasatinib is more active than imatinib. So was there any reason, or can you do? Probably you could consider doing a comparative evaluation of the same formulation.
Yes, actually, imatinib is still the first line drug, but dasatinib is like used for even imatinib resistant cases. So we went ahead with dasatinib, and like in spite of its good activity, the limitation which was limiting it to be used as a first line drug is one problem is this dose. Dose related local side effects it was facing. So we tried, like we made an attempt in in case if we increase the bioavailability, then we can achieve a lower, therapeutic activity at a lower dose, so that we can reduce the side effects. That's why we went ahead with dasatinib. So
Yeah, because these drugs are anyway based on the survival rate of the groups that they are treated. So my understanding is the penetration is better in dasatinib. Probably imatinib can be studied along with it.
yes, sir, sure.
Other thing that I want to know, what is the source of your oil, oleic acid and chamomile oil?
Oleic acid, Sigma, like we procured it from Sigma only, sir. And chamomile oil also we got from Abitec Corporations. Abitec. Yes,
Are they
sir.
certified?
Sir?
Are these certified?
Yes, sir. Yes, sir. The purity grades are also mentioned in it. So we procured those grades only.
What is the purity of these oils?
It was around 99.7 or something, sir, for oleic acid, and for chamomile oil also it was around 98%.
Just a suggestion, it's not a
Yes, sir.
question. Especially these oils are not manufactured generally by known sources. I would recommend these are tested before formulation.
Okay, sir. Sure, sir.
Thank you. Well done.
Thank you, sir. Thank you, sir.
Tarun, you can play the ad till the evaluation is done and we proceed, and Hema can take over.
Tarun, we can add sponsors into the panel. That Parizad is there available and other sponsors.
Sure, sir.
Have you all finished with the evaluation?
बाहर बैठते हैं 2 मिनट या मैंने जाना था जब तुम जाएंगे जब मैं जाऊँ
Priya, can you hear me? Yeah.
Yes, yes.
Yeah, just wanted to mention that I completed my evaluation form.
Okay.
For me, I mean, the ranking is pretty clear from there. And I'm really sorry, but I need to drop off now because I have some time constraints, as I mentioned before. So, but it was very interesting and very diverse topics and presentations. So again, very happy to be part of this event.
Thank you very much. Tarun, can you just confirm evaluation sheet from all the
Samir,
judges?
thank you very much that you joined today also in spite of your personal engagements, and thank you so much for being with us for 2 days. So your certificate I will receive and send it to you.
Yeah, okay, thank you.
Thank you very much. Okay, sir.
A nice evening to all of you, and congratulations to all participants of yesterday and today as well.
Thank you very much.
Thank you, Samir. Thank you, Jit. Bye-bye.
Okay, now I request Dr. Hema Nair to have the introduction of today's judges. So Tarun, can you share the screen?
The evaluation is done, right? It's all over?
Yes. Evaluation done, no, by all?
Okay.
Yes.
All done, Tarun?
Yes.
Okay. Yeah. So this is Hema. And before I take things forward, I would like to extend my greetings to all those present. Both yesterday and today, all presentations were terrific and one better than the other, and they're all winners actually. I'm quite relieved that I could enjoy them, but I'm not supposed to judge them because it would be very difficult to rank them and grade them like that. But let's get to know the persons who have been entrusted with this tough job for today. Of course, as we have all seen, they have been ready with a volley of very pertinent queries, many of which will open up further ideas and research avenues for our participants and tackle lacunae in their work, if any. For past DRPI presentations, we have been listing the judges in alphabetical sequence. Now, we discovered yesterday that all our judges have names beginning with S. So I thought I'd go to the next alphabet, and 3 of them have an A next. And then if we go to the next alphabet, 2 of them have an N next. So I just decided to go random. And it is my pleasure to introduce Dr. Sandeep Tiwari. Can we have the slide, please, Tarunji? Yeah, thank you. Dr. Sandeep Tiwari, who has always been a part of SPDS and all its activities and helped with the evaluation process yesterday as well. So Dr. Tiwari is Head of Technical Services, Pharma Solutions, North America at BASF. He has earned his PhD from College of Pharmaceutical Sciences, Manipal, India, and followed it up with his postdoctoral fellowship from Northeastern University, Boston. While in India, Sandeep worked for Zydus Cadila in the novel drug delivery systems area. Prior to his present position at BASF, Dr. Tiwari was also a fellow, Manufacturing Science and Technology at Teva Pharmaceuticals, Florida. He has also served at Colorcon as a senior manager, global technology development, and then as technical director. Sandeep has been a prolific publisher and has penned 1 book, 8 book chapters, monographs, and contributed more than 100 research publications and conference presentations in the area of dosage form design, new technologies, dissolution science, and excipients. Dr. Tiwari is also the lead editor of the 2nd edition of Textbook of Dissolution Science and Application, published by SPDS. Thank you, Dr. Sandeep. Next, I take pride in introducing Dr. Srinivas.
Hema, yes, you can.
Dr.
Proceed.
Patnala is Director, PHY IND Solutions LLP, Pharmaceutical Consultants and Subject Matter Experts. He's also Visiting Fellow, Faculty of Pharmacy, Rhodes University, South Africa, and Adjunct Faculty, JSS Academy of Higher Education and Research, Mysuru. With a B.Pharm from Bangalore University, Masters in Pharmaceutics from RGUHS University, and a PhD from Rhodes University. Dr. Patnala's subject of interest is pharmaceutical analysis. He focuses on analytical method development in various matrices and QC methods for natural products and associated dosage forms. He's worked in the pharmaceutical industry in quality control, quality assurance, and related R&D departments. In 2010, he joined KLE Academy of Higher Education and Research, Belgavi, as Professor of Pharmaceutical Analysis before taking over his current responsibility. Dr. Patnala is engaged with consultancy services for pharmaceutical quality, research, design, and training. He is appointed as advisory editor to Rule-Tech, Taylor Francis, London, UK. He has published articles in various peer-reviewed scientific journals, co-authored book chapters, and is associated with the WHO Geneva as an ad hoc expert for reviewing analytical monographs of international pharmacopoeia and pharmacopoeial pre-qualification programs. Dr. Patnala has also been with us yesterday, and we are indeed grateful for his willing contribution in raising the bar for our event. It's a privilege for me to introduce Dr. Samir Aduchi, although he had to leave and is not here to listen to us. He's an insider to SPDS, Managing Director, SPS Pharma Services, France. As a chemist, Samir began his career in the analytical development of agrochemical compounds at Sandoz Agro in Basel. During the Novartis merger, he moved to Orleans, France in 1998 to join the analytical group in the technical department, where he became responsible for dissolution. In 2005, he resigned from Novartis to create SPS Pharma Services in Claremont, which is the first and only CRO specialized in dissolution and release testing. And since then, he manages SPS facility and is in charge of project management. In April 2013, SPS Pharma moved to a larger facility and today provides broader range of services to its clients, including routine CGMP testing. The facility has been successfully inspected by US FDA and is registered as pharmaceutical establishment for both US and Europe. Sameer's fields of interest and expertise include analytical method development, in vitro dissolution and release testing across apparatus ranging from USP1 to 7, IVIVC, formulation development, and laboratory automation. Sameer is regularly invited as speaker at international conferences, as well as an expert for various organizations of various scientific societies and health authorities. Thank you, Sameer, for making it possible to be with us for here despite overlap with other commitments. I now take pride in introducing Dr. Stephen Barton. Dr. Barton is Associate Professor in Analytical Chemistry, Pharmaceutical Analysis at Kingston University and a Course Director for MSc in Analytical Chemistry and MSc Pharmaceutical Analysis. He is a chartered chemist and chartered scientist and an active member of the Royal Society of Chemistry. He was chairman of the Southeast Region Analytical Division from 2009 to 2012 and currently sits on the committee as treasurer. He has over 100 publications in the area of drug stability, drug delivery, and pharmaceutical analysis, as well as interests in the effects of pharmaceuticals and other compounds released into the environment. Thank you, Dr. Barton, for pitching in the evaluation process and sparing all this time for being with us, and also for your wonderful questions to our participants. It gives me great happiness to introduce our next judge, dear Dr. Sanket Shah. Dr. Shah is Senior Formulation Scientist, Parenterals and Liquids Development, Pharmaceutical Product Development and Supply, Janssen R&D, Belgium. Dr. Shah is a pharmacist by training from the University of Mumbai and has more than 14 years of research experience in the field of advanced drug delivery. Dr. Shah has diverse industrial experience in drug product development of synthetic small molecules and peptides, spanning pre-NME to clinical stages to life cycle management. Dr. Shah has developed extensive expertise in local delivery formulations with critical insights on drug-device combination products and their impact on in vivo performance. Dr. Shah is also actively involved with academia in innovative drug delivery projects in a public-private partnership funding model. Dr. Shah is co-author of 14 original articles in peer-reviewed journals, 3 book chapters, and has 2 patents in the field of drug delivery and formulation development. Currently, his focus at Janssen is on drug product development of complex parenteral and liquid drug products. Thank you, Dr. Shah. We would like to hear from our judges how they feel about this platform and their overall experience being part of the evaluation process.
We can start with Dr. Sanket Shah. Yeah, I think he's the youngest.
Youngest, yeah.
No, thanks a lot, first of all, for the opportunity to judge. I think the presentations were very insightful, as you already mentioned. And it was indeed a bit tough to score, to be honest. When you need to put the marks, it's a bit of a, you need a winner at the end of the day. So we were put to that process, but it was indeed a very good experience.
Can we have Dr. Barton?
Thank you, Dr. Sanket. Thank you.
Yeah, thank you very much for inviting me to take part in this. It's been very interesting, and as Sanket has said, all the talks were very good, and it was difficult to judge between them. But as we know, there has to be a winner always, unfortunately. In terms of how it's worked, it's all run very smoothly. I was able to listen to all the talks without any problem, and the scoring, once I got used to it, it was very easy, and it's nice the way it's done quickly as well, rather than having to wait for ages. So thank you all very much.
Thank you, Dr. Barton, for that great feedback. We could move on to Dr. Patnala.
Hello to you all once again. It's always a pleasure and wonderful event that I can be associated. There's a lot of learning to, that we take home as what we are, even though we know a few things, but the young scientists are really doing it in a big way, and so happy and to see them, do this kind of high-end work. And truly, this particular event, these 2 days that I have been here, shown me what is happening around the world, and in terms of pharmaceutical research and how the young scientists are really performing at a high level, and the accessibility of instruments and the capabilities have enhanced all these techniques. I'm sure there will be wonderful outcomes which will be beneficial for all the patients all over the world, and also it improves the efficacy of our healthcare systems also. And that's the point I'm looking at, and it's always a pleasure that such fora is provided for the young scientists at a young age and level. So the credit goes to the organizing committee and the people behind it. And I'm grateful and also thankful to all the members of this wonderful academy. And I look forward to further interactions, and also grateful to the judges that I am sitting with and their wonderful insights that they have provided, the learning curve. Much appreciated. Thank you.
Thank you, sir, for that great feedback. Looking forward to seeing you again on this platform in the upcoming years. Dr. Tiwari?
Yeah, thank you, Dr. Hema Nair, for the nice introduction. It has been a real pleasure to be associated with SPDS for a very long time. And every time I join this platform, I think it is very interesting and inspiring to see that the today's generation scientists and the students are really, at least I would say, 5 or 10 years ahead of my generation in terms of the thinking, in terms of their in-depth knowledge. I think that was very evident during the Q&As. So it's always a pleasure, and very happy to see the initiatives from Dr. Shah, Dr. Saranjit Singh, Dr. Ramaswamy, and the entire team, Dr. Padma and others, taking this to a new level and bringing this world level competition. I think next year we should really ensure that we also have a solid participation from Europe as well, to really complete that circle. But it has been a real pleasure to be here interacting with the students, and always happy to support any initiatives, in this regard. Thank you.
Thank you. Thank you so much, Dr. Tiwari. Once again, thank you very much to all the judges for taking time out from your busy schedules. Despite it not being very comfortable time of the day for many of you, since you are in different parts of the globe, your contribution to this event is invaluable and it's gratefully acknowledged. But until you pick the winner, the task is incomplete. So I request all the judges and Dr. Krishna Priya to probably leave this meet and join a separate one, for which Tarun would have sent you some link. Tarun, do you want to add, add some instructions? What should be done?
No, yeah. So we would be leaving the meeting, and I would share the link on WhatsApp group, so all the judges and Krishna Priya madam can join there.
Thank you.
Yeah.
Thank you, Tarun. Thank you.
And Hema, you can carry on with the procedure.
Yeah, sure.
Before we, can I make a comment? I think before we choose winner, I think one thing I can say is women are already the winner of this competition, because all the presentation we saw had women there. So it's already a winner there. Huh?
100%, not 50.
So all the women on the panel applaud what you found out.
But interesting thing is all the judges were men, you see.
Yes, sir. We did note that earlier itself. We did note that. They were all men beginning with the alphabet S. So I don't know, somehow we ended up.
Maybe we should try to change it next year.
Yeah, sure. Yeah,
thank you.
With all the credit and respect to all the judges, just add some more.
Dr. Shah, you are mentioning winners or judges?
Both.
Judges were
Both.
Yeah.
Okay.
The winners, I don't know.
Judges to please
I'm not evaluated, so I can just talk only about the judges, because previously it was mentioned that all the presentations were by women. So I said all the judges were men. That's all.
For once, they can get judgmental, I guess. So, yeah, let's proceed. And in the meantime, while the judges are doing the tough job of getting to record the winners in a serial manner, let's do some more thank yous before we start with, we reach the stage where we can announce the awards. While we had our global chair, Professor Saranjit Singh, and the regional chairs, including Professor James Polly, Professor Matthias Wacker, and Professor Kamal Dua speak to us yesterday, we would also like to acknowledge the inputs by the vice chairs and all the other organizing team members, some of whom have worked relentlessly to set the ball rolling and to ensure good participation from their respective regions. The vice chairs who have been part of this event include Professor Hardeep Saluja, Professor of Pharmaceutical Sciences, Bernhardt Scholar, PharmD MBA Program Coordinator, Southwestern Oklahoma State University, USA, who was the Vice Chair, USA, Professor Atsushi Kambayashi, Associate Professor, Faculty of Pharmaceutical Sciences, Tokyo University of Science, Japan, who's been the Vice Chair for the Southeast Asia and Japan region, Dr. Liefeng Kang, Senior Lecturer, University of Sydney School of Pharmacy, Faculty of Medicine and Health, Australia, who's been the Vice Chair for Australia and New Zealand, Dr. Dayardi Diarsi, Associate Professor, Pharmacy and Pharmaceutical Technology, Trinity College, Dublin, Ireland, who is the Vice Chair for Europe region, and of course, the Vice Chair for India, Professor Krishna Priya Mohan Raj, who just left this meet to sort out the results with the judges. Thank you, all of you. And if any of you are present over here, we would be really happy.
All are present.
Oh, great.
So you can take one by one.
Dr. Saluja, we would like to hear from you. Your thoughts and experiences during this DRPI journey so far, and any plans that you may have to share with us.
Okay.
Thank you, Dr. Nair. Appreciate. So when I was thinking about what I'm going to talk about, the first thing was how I'm going to greet you all. Is it good morning, good evening, good afternoon? So I'm going to keep it simple. I'll just say greetings from USA. First and foremost, I must commend the exemplary presentations we have witnessed over the past 2 days. I wish to congratulate all the participants for their outstanding efforts. The depth of knowledge, innovation, and dedication displayed is truly a testament to the talent of our community. My name is Professor Hardeep Saluja. This year, I had the distinct honor of serving as the Vice Chair for the DRPI US Committee. As we all know, this was the inaugural year for the USA as the DRPI unfolds. It has been a journey of learning. We received an impressive 23 abstracts from the USA, and notably 22 were in the PhD category and 1 was in the master's category. Such participation speaks volumes about the interest and commitment in our community. While this year laid a strong foundation, next year we aim to expand and grow even further. I'm sincerely grateful to Dr. Vinod Shah for affording me with such a wonderful opportunity. Earlier this summer, I had the privilege of attending DISO India conference in Mumbai. And I must say my experience was wonderful. Thank you for the hospitality. Enjoyed meeting with everyone. And the dedication and caliber of research there left actually a very profound impact on me. I also owe my heartfelt gratitude to Dr. James Polly for chairing the DRPI US with his expertise, and Dr. L. Ramaswamy for his steadfast leadership throughout. I have to say here that Dr. Ramaswamy should sleep, because I don't think so he sleeps at all. Doesn't matter whether it is evening, morning, or whenever you text him or email him, his reply is always there within few hours. As we wrap up today's event, let's remain inspired and continue to push the boundaries of what we have achieved. Here's to many more years of innovation and groundbreaking research. On a different note, if you are attending AAPS conference this year in Orlando, I would like to extend an invitation to all of you to attend American Association of Indian Pharmaceutical Scientists Banquet, which will be held on October 24th in Orlando, Florida. I'm serving as the president for AIPS. I would love to meet you all. I think these conferences gives us a platform and opportunity to actually meet face to face. Thank you all for an unforgettable DRP event and my best wishes to all of you. Thank you.
Thank you, Dr. Saluja. That was great encouragement for us from this side. And also thank you for the extension and for the invitation extended to the meet at Orlando. Thank you, sir. Looking forward to seeing you again next year. So I now welcome Professor Kambayashi. Sir, you could just unmute or switch on your video and join us.
We can't see him.
Acha, okay. Maybe, I don't know.
Dr. Kang is there.
Yes, Dr. Kang is there, Hema.
Yeah, okay. Dr. Kang, could we hear from you? How you found it this year and what are your plans for the coming times?
All right, can you hear me well, everyone?
Very sure, yeah, we can.
Okay. Hi, I'm Li Feng from the University of Sydney, Australia. Thank you very much for giving me this opportunity to say a few words. So first of all, I'd like to say the event is very well organized with a great support from the organizer in many ways like email, calls, and also has a lot of company sponsors, which is wonderful. And also enthusiastic participants from all over the world. And the process is very well timed so that to accommodate all the participants from every corner of the zone, different time zones. So I'd like to also thank the organizer for inviting me to attend this competition. Also, I thank Dr. Cam Dra from University of Technology, Sydney, Australia, for inviting me to become a vice chair in this Australia-New Zealand scientific committee. So lastly, I'd like to congratulate everyone who has been involved in this online competition, which is really amazing and rewarding, and look forward to participating in next year. Thank you very much.
Thank you, Professor Kang. Thank you so much. Okay, actually, Professor Kambayashi has a message here. He's also raised his hand. He says he cannot put on his audio and video, and unfortunately, Tarun is also not with us at this time. So
we can take when
a little later, yeah. So that's
ask him to insert him into the panel.
Yeah, Professor Kambayashi, we need to just wait for a few minutes. Once our tech person joins us, we will request him to make the necessary changes and then hear from you. Thank you, Professor Kang, once again. Looking forward to seeing you again. Thank you so much for all your enthusiasm, and in this kind of spirit, I'm sure we can go a long, long way. Going further, this event, the prizes for all the winners, all related arrangements could not have reached this level without the backing of our sponsors, SOTAX, BASF, and ACG. I now invite the prime force and the dynamic person who has been in the driver's seat for SPDS and all its events, and of course DRPI also, Dr. Lakshmanan Ramaswamy, General Secretary SPDS, Managing Director SOTAX India Private Limited, to welcome the representatives from the sponsors who are here with us, and also to take the event forward by inviting our chief guest, Dr. Vinod Shah, as we begin with the steps towards the much awaited culmination of this grand finale, announcing the winners of DRPI International 2023. Maybe, sir, after the sponsors, we could see if Tarun is back, we could have
Sure.
Professor Kambayashi before we proceed.
Absolutely, we need to listen from him. Thank you, Dr. Hema. It's a wonderful conducting the event today. My once again, good evening, good afternoon, greetings to all. First, there are two platinum sponsors, the SOTAX and ACG. And we have today the presence of Mr. Ajit Singh from US. And again, he is in the, not has come to the panel. I think Tarun has to take him to the panel, then only he will be able to speak. So Kedan, are you able to speak? Because I could not see their name.
No, they're not
okay.
there, sir.
No problem. Then maybe Tarun will be coming soon in the next 2-3 minutes. In the meantime, Krishnapriya called Tarun on mobile and just ask him if it is okay to take 2 people. We have to take into the panel immediately.
Yeah.
And one is our doctor from Kambayashi from Japan, and Mr. Ajit Singh or Kedan Parman's mobile is being used. We have to take both of them to the panel. In the meantime, can I request the business head of South Asia, BSF Pharma Services, Mr. Satish Murkat. Satish Murkat, besides his official designation, he has been very supportive to SPDS as well as for DRPI, since he has come to know that we are doing such program. So let us listen from him how he feels as head of Southeast Asia, BSF Pharma Services, and how he would like to further support these research events of SPDS and DRPI. Yes, Satish, can we invite you, Satish?
Yeah, Doctor. Can you hear me?
Yes, Satish.
Yeah, greetings to all from my side, and thank you, Doctor, for this introduction. And first of all, I should thank you for introducing the SPDS and activities to us, and then we have been part of it since last year as a sponsor. But prior to that, also my colleague Sandeep Tiwari has been associated with SPDS in a big way, and he's been a judge today also. So thanks to Sandeep also. So I take this opportunity to thank everyone for a great event, and it humbles me to be part of the scientific sessions. I mean, a lot of interesting presentations, and as Sandeep also mentioned, it's all very, very high class ones, which probably is much bigger than at our times when we were in the pharma world, in the pharma studies. Yeah, so that's the quality of the things. And of course, this is also being presided by visionaries and leaders in the pharmaceutical industry and the pharmaceutical analysis. So that itself speaks a big volume about the event. On behalf of BSF Pharma Solutions, actually, I'm thankful to the organizers and also congratulate the organizers, SPDS, AAPS, APTI, and all others, to take it from an event which was running to an international event today with participants across the world in a seamless digitalized world today. It's not an easy task. It's certainly one of the great events of such kind where scientist community or the young scientists from across the world are joining together in a common platform. And of course, all the judges who have been patiently listening to everyone, asking the right questions and encouraging all the participants, and all the participants across the world whose work has been exemplary, and they have been courageous enough to present this across to the entire world. And finally, winners will be announced soon, so congratulations to them in advance, and that is going to come very soon. Only just a brief about BASF. At BASF, we create chemistry for a sustainable future. That's what we are into. And we support innovations and creativity in the scientific world. And that is exemplified by the fact that more than 10,000 scientists work in the R&D across the world with a budget of more than 2 billion euros. And just in 2022, more than 1,000 patents were filed. So that speaks volume of the BASF's commitment to scientific community. And in BASF Pharma Solutions, we are responsible for developing and supplying ingredients, especially for the functional excipients and selected APIs within BASF. And we have got more than 860 to 60 excipients, and I'm so happy to see that a lot of the presenters had used some of our excipients to create a differentiation in their formulations. And our commitment is to support the scientific community right from the student life to the experts on whatever they want in terms of material, technology, and any other support that may be required. So that is our commitment, and we are very happy to be associated with SPDS and DRPI, and will continue to work together to make this even more bigger and successful. I won't take much of your time. Thank you very much once again. Yeah.
Thank you, Satish, for your nice remarks. And I remember still when we briefed you for the first time, you're a little skeptical. However, when we said that we are going to work with students, especially with researchers, and you have shown a lot of interest and you are part of nurturing. Today, DRPI Global Serial is the first episode. You go back and see any serial in the world. In the first episode, you don't have much viewers or much advertisers. There will be few viewers, few advertisers. But when the episode goes to 10, 20, 30, the momentum gains up. Then you will have more advertisers and more viewers. But in the case, a research presentation like this, I feel 10,000 people should see, because the efforts of 200 plus researchers who have put, if you calculate the number of hours, days, weeks, and years, nobody can value that. So we are all very fortunate to sit in our own chair, in our own dining room, in our own bedroom, in our own home or office, to watch on a weekend such a beautiful research presentation which has happened across the globe. I am sure the pharma industry and academia will come to know about this in the due course of time. And I don't mind, I am not exaggerating. Next year, we will have minimum 100% more presentation than this year. And we would like, again, these three sponsors to continue to promote and support this event globally, when in spite of its growing year after year. So thank you once again, Satish. Your support at this early stage is very valuable. And we record our appreciation on behalf of SPDS and DRPI organizing team. Thank you once again, Mr. Satish. Now we have a person waiting eagerly to talk to us, to address to us. And he's always been a well-wisher of SPDS and a patron from the day one. And any scientific organization, he is behind. And that is none other than the chairman of ACG, Mr. Ajit Singh, who is also the chief patron of SPDS. I would say the founder patron. So may I request Mr. Ajit Singh to address now this audience and these researchers who are keenly waiting to see their results. Please, handing over to Mr. Ajit Singh now.
Thank you, Dr. Ramaswamy. It is my pleasure and honor to be able to talk to you for a few minutes. I remember the days just about 10 years ago when a gentleman called Dr. Ramaswamy had an idea. In his mind, it was a big global idea, but we had to start small. He was joined by a man called Dr. Vinod Shah, incredible personality, former head of the American Association of Pharma Scientists, the first from India, the scientific secretary of the FIP, which we should all take much more interest in. It's a great organization. And from there, with incredible dedication and work and reach, SPDS is becoming close to the stature of probably the most interestingly widespread organization in the scientific area from India in the world. I was particularly interested to see that in our DRPI event, we have many award winners from many different countries. And I think this is the first example of a developing country giving birth to an organization that is now giving global recognition, assistance, and scholarships. As a part of the family of SPDS, I am very proud to see how this scientific organization is developing. While we were mostly focused on research all these years, it is now the wish of the committee and the organizers to shift SPDS from mostly education, which it was doing such a fine job in, to now the wider area of research. And I do wish it all success in this ambitious feature. You know, India is supposed to be a sort of developing country, largely having developed, but people have not recognized its incredible power, strength, and research strength in the area of pharmaceuticals. It's reached the status of the world's largest manufacturer of pharmaceutical generics. It supplies 40% of the requirement of the most advanced countries of the world, and therefore it is a fit place from where research should be launched and coordinated around the world. What India does today, there are about 50 other developing countries that follow its lead, whether it's in quality control or it's in education. India plays a very, very significant role. Today, the DRPI of the SPDS is joined with the joined by the Association of Teachers of India, also by the AAPS, and of course ACG's SciTech Center. We are all very proud to be an integral part of this event. I have a request. Now that all of you young students and researchers in industry are part of the family of SPDS, please make an attempt this year to bring in 2 more members. If there is anybody in the world that can advance the membership of this association, it is you and not others. So please use your talent and your contacts to get this done. I am glad to be involved with SPDS. I believe that each one of us makes an impact on the world. And the impact that you make depends on you. You determine your impact. And to the extent that the SPDS and all its associates are able to assist you with research and with education, please do take advantage of it. And let's move ahead together. Thank you very much for listening to me.
Thank you, Mr. Ajit Singh, and thank you very much. I must take 2 more lines to speak about Mr. Ajit Singh. My association starts with my first meeting in 1990s at Bombay. I see last 32 years his journey. Today, he is a living legend in pharma industry, not only in India, but also in many other countries. So we all should pray, in spite of some personal difficulties, like a week back, a surgery in his right hand, a few days back, a pacemaker change, traveling from Budapest to Bombay to New York. In spite of that, he is able to come and address today. That shows his commitment. Like today, we have Samir, we have Jim Polly. Everyone has personal issues, but in spite of that, I used to put, if I am in that position, will I be able to go and speak? This is the question I asked. So these 3 gentlemen plus many more, we are committed people. That is the strength of SPDS and DRPI team. As Mr. Ajit Singh has rightly said, that next year, each one of us, a world over, who are working for SPDS or join part of SPDS or DRPI, can we take a little extra mile to get 2 or 3 more participants in the research presentation next year, so that we have more than double the number. Just to add to that, I counted, we have more than 500 good universities in the world doing pharmaceutical dissolution research. So maybe 100 are in Europe, more than 100 are in US, and we have around 150 plus in India. So we can tap all these together, plus rest of the world, in like Singapore, everywhere we have 20, 30. We can aim for 500 research presentation for next year to get through DRPI. And that will be a great inspiration for those 500 students, researchers, or young researchers, late researchers, and their guides. And let us motivate them, invite them for presentation, give them the opportunity, give them the dais, give them the stage, so that their name and fame will go world over, and we will learn from their research, sitting at our own table or chair, wherever we are. This is my dream. I'm sure we will work towards that, sir. Thank you very much for being with us. I would like to see you fast in Bombay and discuss about our next year, because Mr. Ajit Singh has given his office as our address. SciTech Center is our registered office address, and he has also shared whatever support SBDS needs, please come and tell. So I take that liberty, but I don't misuse that liberty. Selectively, I use their resources for running of SPDS. So thank you very much, sir. Thank you very much, Kedan, for arranging Mr. Ajit Singh today to us.
Thank you.
Okay. Now we have Asushi Kabayashi has joined from Japan, who is our vice chair, along with Dr. Matthias working together. We had a good show. So we will listen from Dr. Asushi, how you felt being part of this team and seeing this event yesterday and today. Yes, handing over to you, Doctor. Please unmute. It's muted. Unmute. Yes. No, I think the microphone.
So can you hear me?
Now, yes. Now, yes. Come closer. It's working. Yes.
Okay. So good morning.
Great.
Good afternoon, ladies and gentlemen. I'm
Come closer. Come closer to the microphone. Yes.
Introduction that I'm also a director of DRPI and Southeast Asia. So as you may know, the we organized Japan and Southeast Asia DRPI meeting last August. It was very sponsored by Professor Martin. Hi, Matthew. So I think that was a very successful event, and I also think one of the that development and the so I hope this DRPI meeting good medicine for that's all. Thank you so much.
So our judges are back. I think they're joining back. The job of evaluation is done.
Yes.
Yes,
And
yes.
we will hand over the stage back to Dr. Ramaswamy once he's free. Dr. Ramaswamy, back to you.
Thank you. Thank you. Okay, I think unfortunately, Dr. Kambayashi's talk, we could not listen much, maybe some issues in the internet. Nevertheless, thank you very much for Asushi, and I want to once again thank all the global chairs and vice chairs. Without this, their efforts, their time, their involvement, this program would not have taken place. So we all have to give a big hand to all the global chairs, vice chairs of various regions. And next year, it is them who are going to make it better or a great event than this year, which I am very confident of. So once again, thank you, and thank you to them. Now, coming to an important, I think all judges are back. And Tarun, are you back, Tarun?
Tarun is compiling, making the certificate.
Okay, fine.
Getting the check ready.
Okay, so let us leave him. Let me see how I can continue this session. Fine.
So we have, sir, have we had Dr. Vinod Shah's?
Yeah, wait a minute. Before getting into Vinod Shah, I have one more job to finish. We listen from ACG and BSF, and from SOTEX, except me, nobody else is there. Of course, Samir was there and he also left. So let me take this 2 minutes to speak about from the part of SOTEX as a managing director of SOTEX, who is the prime sponsor. Right from the start, we are fortunate enough that we were one of the platinum partners in every event of DRPI or SPDS, and we were instrumental in setting up this organization, which you will see in the formation day. Mr. Rolf Benz and John Louis Ratton were in India, along with the team who formed in that Vijay Shirsagar, Abba Doshi, many teachers were also present in that meeting at Westin on 26 January 2012. From that commitment, and why SOTEX got into it? Yes, they are the leaders in dissolution science, dissolution testing, as well as in the services provided to world over for pharma services. So besides bringing instruments and software, SOTEX is also committed to promote the research and development in the field of dissolution cellulose. With that objective, the SOTEX is always been encouraging SPDS initiative, and I got full support from the SOTEX management and my team in India and abroad to spend not only my time, efforts, thinking, strategy, however, to execute most of the events with their help. So with this, I would like to thank all SPDS trustees, executive committee, DRPI, scientific committee, DRPI core committee, DRPI global chairs, vice chairs, for giving us this opportunity to participate in this great event as a platinum sponsors. I record on behalf of SOTEX my sincere thanks as well as best wishes to all the initiatives of future of SPDS and DRPI. Thank you. With this, we go to the next session. It's a great honor for me to speak about a person who has been, I will not say instrumental, who has been the backbone of our SPDS activities right from start. So I thank God that in 2013, God made me to meet a gentleman, namely Dr. Vinod Shah in Bombay. So that first meeting in a scientific conference, I had a chance to talk about, sir, we are going to have a society called SPDS. So he showed so much interest when I heard the word dissolution. Amazing, amazing. I could not explain that experience I faced. Since then, till today, we have been associated in many ways, through email, chat, talk, conference, meetings, and his heart always beats for dissolution. So let me have a pleasure of introducing him formally. Is this slide visible for you, or it's only for me? It's
okay. Not visible.
So you'll have to share your screen, sir.
Now?
Yeah, yes.
Yes.
So this is the same magic Dr. Vinod Shah did for dissolution, which we were not aware of it. So we all made it clear through his research. So let me have the privilege and pleasure to introduce Dr. Vinod P. Shah, pharmaceutical consultant from USA. He is the founding president of SPDS US also. Dr. Shah is a gold medal distinction pharmacy degree holder from Madras University, India, and PhD in pharmaceutical chemistry from University of California, San Francisco. Dr. Shah worked as US FDA from 1975 to 2005, 30 years, 3 decades, where he developed several regulatory guidances for industry in the area of dissolution, SUPAC, SUPAC-SS, bioanalytical method validation, topicals, bioequivalence, and biopharmaceutics, and pioneered method development for in vitro drug release for semi-solid dosage forms. He is a recipient of many FDA awards. He is author, co-author of more than 330 scientific papers and is co-editor of 4 books, including the Desk Book of Dissolution, Science and Applications, released by SPDS. Dr. Shah was scientific secretary during the period 2003 to 2011 of International Pharmaceutical Federation and president of American Association of Pharmaceutical Scientists in 2003. Dr. Shah is a recipient of FIP Lifetime Achievement Award in Pharmaceutical Science, IDMA Eminent Pharmaceutical Analyst Award, India, honorary doctorate from Semmelweis University, which is from Budapest, Hungary, which is a 250 years old university. Honorary doctorate from University of Medicine and Pharmacy, Carol Davila, Bucharest, Romania. SPDS Award of Excellence, Lifetime Achiever Award, AAPS Distinguished Pharmaceutical Scientist Award, and AAPS Global Leader Award. Dr. Shah is the international chairman, as I mentioned, Society for Pharmaceutical Dissolution Science from 2012 till now, and president of SPDS US chapter from 2019, from the formation day till now. So with this brief introduction, on behalf of all the dignitaries present and the research students, research presenters, winners present in this forum, we invite Dr. Vinod P. Shah to deliver his address to us today. Yes, Dr. Vinod Shah, please.
Thank you, Dr. Ramaswamy, for a very extended biography of my background. I would say that greetings to all. And today it's like a dream come true, a reality. It is my great honor and pleasure to address the dissolution research scholars, their mentors, judges, and professionals from all over the world at DRPI-World 2023 Grand Finale event. Let me just check. Are you able to hear me properly?
Yes, sir.
Okay.
Yes.
To welcome everyone at this event is really like welcoming a dream come true. This is a flagship event of SPDS, one of its kind in the world, for the young pharmaceutical research students and scholars in the area of dissolution science. Another flagship event of SPDS is the DISSO India, held every year since its inception. SPDS, the Society for Dissolution Science, is a unique and only society in the world solely dedicated to dissolution science. It started in India about a decade ago, but has now gone globally with the first chapter established in the US. The DRPI is the first and the only global event for dissolution scholars to showcase the research in dissolution-related fields. My heartiest congratulations to the SPDS and the DRPI team for these events. I'm proud to be associated with both SPDS and DRPI. The creative and the inspirational brain behind these events is that of no other than Dr. Ramaswamy. It is true that such creative enterprise always needs a motivated and dedicated team. For SPDS, the team included, but is not limited to, because I cannot remember all the names, but some of the names includes Mr. Vijay Sirsagar, Dr. Padma Devarajan, Dr. Subramaniam Vaidya, Dr. Mangal Nageshwaran, Dr. Arvind Bansal, and the others. And for DIPI, it included Dr. Saranjit Singh, with Dr. Ramswamy, along with Dr. Mala Menon, Dr. Krishna Priya, Dr. Hema Nair, Dr. Varsha Pradhan, and the IT expert, no other than Tarun Soni. Together, they organized and implemented the DRPI event, first of its kind in India and probably in the world. A unique, innovative, and interactive platform was developed by the IT specialist Tarun Soni to evaluate the abstracts and the delivery style by team of experts. The entire event was completely digitalized. DRPI is a fine way, in my way of thinking, it's a fine way of training the students for the future role and an excellent way to promote and recognize young researchers in the area of dissolution science. Now, after 3 years of success, DRPI has become global. In other words, the DRPI, which was standing for India, has now been transformed into I for International, DRPI International. The DRPI event is co-sponsored with the most prestigious pharmaceutical association in the world, American Association of Pharmaceutical Scientists, AAPS, that really stands and indicates the strength and the quality of SPDS as well as for the DRPI. The success of DRPI global event is due to the great enthusiasm and support and the hard work of the global chairs and vice chairs, namely Dr. Saranjit Singh and Dr. Krishna Priya from India, Dr. James Pauly and Hardeep Saluja from USA, Dr. Nicholas Fodaki and DRG from Europe, UK, Dr. Matthias Walker from Singapore, Dr. Atushi Kambasi from Japan, Dr. Kamal Dua and Lifeng Kang from Australia. All these leaders have played a very vital role in organizing country-wise competitions. My many thanks to all the chairs and the vice chairs, and I am hopeful for their continued support for the future growth of DRPI events 2024 and beyond. I have listened to all the research presentations of both the days. They represent to me the cream of the crops. All were excellent. And in a sense, they are all the winners. And I would like to thank all the judges for critically evaluating all the presentations. And as we heard from some of the judges, it has been very difficult to score first, second, and third. And that's why my feeling is all presentations, all the presenters are the winners. In the end, I would like to say that it is important to recognize and acknowledge that DRPI is a very unique platform which encourages the young researchers to showcase their scientific work and talent in a global competition. It's a great platform. Thank you, Tarun, for creating such a wonderful platform where the young researchers can utilize it to show their talents. I would like to thank you all for listening to me, and it was a great experience to follow for the 2 days the events of the DRPI. Thank you all. Back to you, Ramswamy.
Thank you very much for your vibrant address, I must say, at this juncture. And every words, lines mentioned by you have gone to our heart. Thank you once again. At this point of time, I think it is appropriate for me, on behalf of the full team present here and some who could not present, from the SPDS chair, that we have decided yesterday, based on the suggestion of Dr. Saranjit Singh, that September 2nd will be celebrated from 2024. 2nd September, am I right, sir?
Yes, sir.
2nd September will be celebrated as the International Dissolution Day world over. So there is a major significance for this day. That is the birthday of the living legend on dissolution. That is none other than Dr. Vinod P. Shah. And yesterday, when I wrote to few university professors whom I have some contact across the globe, amazing, amazing. Everyone has made a record, and in their university calendar, in their university functions, they are going to celebrate next year onwards, September 2nd, as the International Dissolution Science Day. It's not International Dissolution Day, it is International Dissolution Science Day. So let us give an honor, let us give a big hand for everyone, and we are declaring this, and we will officially pass this resolution in our AGM, which will be scheduled on 30th of September in Bombay online.
What is that all? Not International Dissolution Day, but International Dissolution Science Day. You have to clarify. Because International Dissolution Day can mean a lot of other things also.
Okay, so we go ahead now with this. Is Tarun is back?
Yes, Tarun is back and results are ready.
Fantastic.
Could you unveil
the abstract book?
Okay, I have to close. I have to, what do you call?
It's done, sir. It's done. Don't worry.
Move out,
Tarun
no?
Tarun will do the needful, sir.
Tarun, we have to release the abstract book with Dr. Shah.
One minute.
So sir, your screen
Tarun?
sharing is stopped. Yeah, your screen sharing is stopped, sir. So don't worry about it.
Thank you. Now the next important event of the day is that we have made a nice compilation of all the research presentations made for DRPI 2024. So we request Dr. Vinod P. Shah, our chief guest today, to unveil this compilation of the very, very interesting compilation, and all of you will get an electronic copy of this compilation, which is the scientific abstract book, which has got messages from many leaders of the SPDS and DRPI, and also the abstracts which has been presented in all over the regions. Am I right, Tarun? All the regions abstracts are there in this.
Yes, sir. Yes, sir. All the regions.
So we request Dr. Vinod Shah, you just click your enter, it will open. Your computer, you touch enter, this will open. Tarun, you take over. And we'll show what are the contents in this. Yes, thank you, Dr. Vinod Shah.
It's a great pleasure to open this abstract book. Thank you, Ramaswamy.
Yes, it's the first of its kind in the world now. So you will find in the first, second page, the what you call in the serial, the particulars are there. Initially, few pages are covered with the messages from Dr. Vinod Shah's message is there, as well as his Dr. Saranjit Singh, the profile as well as the message, a brief profile and the message. Mr. Vijay Shirsagar, our founder president, profile and the message. Yes. Padma Devarajan, Dr. Padma Devarajan, our current president, his profile as well as Dr. James Pauly, his message is there. Profile we can add. Dr. Kamal Dua, his profile as well as his message. Very nice. Dr. Munira Momin, who is the current scientific chair, her profile and message. Dr. Mala Menon, her profile and her former vice chair and the trustee of SPDS since start. Her message is there. Dr. Hardeep Saluja, his profile as well as is the co-chair of US SPDS DRPI. Dr. Life Kang, his profile as well as a small short message. Dr. Krishna Priya, the Vice Chair of DRPI, and their profile and message. Dr. Varsha Pradhan, her profile and message. She is one of the core committee member. Of course, my message is already read today in my address today. These are all from India. Now I think the abstract starts. If I start reading, the whole day, night will go. So I'm not reading this. You just skip over. Everyone will get a copy. How many abstracts, Tarun?
67 from India, and then we have from US about 20 abstracts, 21 abstracts.
100 in all, 100 in all, exactly.
So we have 100 plus abstract, which will go free of cost to all the research students, teachers, guides, whoever wants it across the globe, a soft copy. Yeah, SPDS is or DRPI not going to charge anything for this.
US from Japan and Southeast Asia, and one abstract from Australia.
So it will be a challenge to all. I could challenge that you will not find any abstract summary book like this anywhere, any part of the world. It's a great compilation, Tarun. Thank you very much.
Thank you.
And in the end of this book, this is totally how many pages now?
147 pages, sir.
145 pages. Once you really print and bind, it looks very nice. Of course, we have the sponsors and some pictures of our earlier events. Rajiv Raghuvanshi, you will find many leaders from pharma industry and regulators participating in our programs, right from AAPS, APTI, government, FDA, USP. So there is a first announcement. Tarun, thank you for that. We are happy to announce that DRPI 24 is already being planned. 24 activities will begin month of April 24 with a call for abstracts. So once we are ending today, and this is going to be an end of a good beginning for next year. So we will make a better strategy, better planning, so that from 500 universities next year, can we get at least one abstract each for the regional as well as the competition. And from there, we take the global competition 2 or 3 for each stream, depending on the continents which are participating. I'm sure next year from Europe and UK, we will get good number of abstracts because the teachers have started writing, we want to participate in DRPI. So we will work, and everyone has got contact in Europe. So let us utilize our contact from tomorrow to write to the teachers in Europe and UK and try to mobilize the abstract from that continent also. Okay, thank you very much. And at this point, before saying, we have Mr. Vijay Shirsagar with us. Vijay, are you there? Can you unmute? Can I?
I'm there.
Yeah. Can you please give your feelings now, even though 10 years is over, you are the ex-president emeritus, I would say. How do you see this event this year and your feelings, please?
Rama, it's a fantastic feeling of extremely high amount of joy and pleasure. And very glad to see the, the quality of the research from developing country like India, apart from developed countries. And it is going to be a big blessing for the mankind if all this research turns out into a commercialization. Even if I say 25% of them turn into reality, it will be a big blessing for the humanity. And we are very keen to see such products going from developing countries like India. And that's where we industry people have been harping on for quite some time, because we have played enough in generics and we are the leaders there, no doubt, pharmacy of the world. But somehow this element of giving it back to the humanity back to the world. I think that's where we have not done well so far, but with this kind of research which is being initiated, compliments to all of you. I think Dr. Saranjit Singh, who actually conceptualized it, and Dr. Rama, our great motivational factor, great creative factor, having taken it forward. And of course, blessed by none other than Dr. Vinod Shah, and very expert and dedicated team. Whether I think if I start taking names, some names, important names will miss out. But the way I think Dr. Krishna Priya, Dr. Hema conducted it, it's very high quality. So I think thank you all of you, and great support from SPDS, all our members, including Dr. Padma and of course Dr. Samir, who has been not actually SPDS but Sotax. And I must compliment that Sotax is one company which is right from day one, I would say. And it has been a big support behind us. I think without that support, we could have not moved to the extent which we have moved today. So thank you. And some important names I missed out, but my heartfelt feelings are there with all of you, and excellent job done.
Thank you, Mr. Vijay, for your continuous support in many ways. And we have new projects coming up in SPDS. We will continue to have your support. I'll come back to you for the other people. In the meantime, the results are ready. A lot of students are waiting to see. A lot of guides are waiting. So Tarun, can we start now? So we will go with the third prize winner of
early stage researchers, sir.
Okay, we call it as early stage registrars, means Masters, MPharm and PharmD included. So let's see, this final happened today since morning, and it's quite unfortunate that we had only 4 contenders out of 5. One could not join. Maybe next year we will have 10 to 12 minimum contenders for Masters also. Nevertheless, the presentation was excellent. All the 4 presenters, not only this 4, even earlier, the Masters who presented in their local competition was also excellent research, excellent research. Good work done by them. Congratulations to everyone. End of the day, few winners, 1, 2, 3. Let's go with the third, rather second runner-up, Tarun. Can you show? The second runner-up is Bharti K from Naipur, Ahmedabad, India. Congratulations, Bharti, for your excellent job and excellent presentation. And you deserve this prize and the certificate. You deserve the third prize. Once again, please go back, Tarun. The title of her presentation was Formulation Development and In Vitro Characterization of Hot Melt Extruded Cyclosporine Ocular Insert for Dry Eye Disease, guided by the supervisor, Professor Derajaram Beniwal. Our sincere congratulations to Professor Derajaram Beniwal, and you can feel proud that one of your student researcher got this globally acclaimed, the second runner-up position. Best of luck, Bharati, to you for your continuous work. Remember the area which you have focused. Every human being is suffering from early days nowadays with the invent of TV, computer and mobile. You will see 50% of the students in the college are wearing glasses nowadays. So you can imagine when they grow old at the age of 40, a great percentage will have eye issues. So I'm glad that you have focused this core area. Continue doing this. And if any support you need in the area of dissolution further, feel free to contact SPDS or even SOTEX. We will be there to support you. So can we request the third prize to be handed over by the sponsorer, BSF? So can we invite Satish, who is the business head of South of Asia, BSF Pharma business, pharma, sorry, BASF of Southeast Asia, Pharma Systems. Please, Satish.
Yeah,
You are
thank you,
there, so you can
Doctor.
hand over. And can we also request Dr. Padma also to join with Satish and hand over the certificate as well as the cash award. The cash award is $1,000. Bharti, in your this will be the first US dollar you are earning, and I'm sure you will earn more in dollars or euros in your future career.
And Bharti has moved to US for her further education.
Fantastic. So is Bharti online now?
I'll check, sir.
If it is there, get all the students ready so that we would like to give a minute for them. If they come on stage, would be better.
I think we can have everybody in now.
Yes, all the people in the, especially the presenters, all the 5 presenters, and of yesterday also they can be in the.
So Bharti is there on the panel now.
Yeah, Bharti, can you listen me? Can you come with your video?
Yes.
Fantastic, Bharti, how you are feeling now to see the certificate and in front of all these luminaries and $1,000 check?
Yeah, I'm extremely grateful for this opportunity. It's been a really tough competition through various rounds, and it has given me a lot of exposure. So thank you, SPDS and the AAPS, for such an amazing opportunity. And at this moment, I'm like very grateful to my mentor because he has supported me throughout this journey. Yeah.
Perfect, perfect. This certificate will take you, add a lot of value to you. Look at the people who have signed. I am sure no one else in the world will have a certificate like this. Okay? Yeah. And I'm sure you will consider the certificate is more value than this check. So I wish you all the very best on behalf of SPDS and DRPI team, and continue your research. Don't stop it. Okay? Best of luck to you.
Thank you.
Yes, thank you. Tarun, we move to the first runner-up of the young researcher category. Here, the first runner-up is Ms. Darshini M. from National Institute of Pharmaceutical Education and Research, SAS Nagar, Punjab, India, Naipur, Mohali. That's what known, famous. And very nice. Congratulations, Darshini, for your excellent work. And you are the first runner-up. You are the second prize winner. The abstract title, Super Saturable Bioself Nano Emulsifying Drug Delivery System for Improving Oral Bioavailability of the Dasatinib. And the guide, none other than Professor Sanyog Jain, who is our Vice Scientific Chair of SPDS also. So thank you very much once again for participating. Our hearty congratulations, Darshini. Can we request Dr. Munira, is he there? No. Okay, then we request Dr. Padma only again hand over the certificate. And Dr. Farizad, are you there from ACG?
Yes.
Hello, Tarun.
Yeah.
Yeah, Dr. Farizad. Thank you, Farizad, for joining. You can on your video if possible. And please hand over the cash prize, which is sponsored by ACG.
Yeah,
Yes.
Dr. Parisad, please do the honors.
Yes,
Yeah.
Dr. Farizad.
Congratulations, Darshini, and wish you all the best. Is she around?
Darshini, can you come on the panel with video?
Yes, sir. Yes, sir. I'm here.
Fantastic, Darshini. I could see your face. You are so happy today, and you can and you should be. Thank you very much for participating, and you have come up through various stages, and you did a very good job today, and good presentation, good topic, good research. So here is your prize of $1,500 for the first runner-up, sponsored by ACG. So Dr. Parisad will arrange to send you that. Thank you, Dr. Parisad. And Darshini, anything you would like, and who is your guide?
Yes, sir. My guide is Professor Sanyog Jain,
Oh,
sir.
Sanyog Jain, Sanyog Jain.
Yes.
Yes. So
Yes,
first
sir.
we listen from you, and then maybe we could listen from your guide also if he's present.
Yes, sir, sure. First of all, thank everyone because it has been a wonderful platform and in fact a very, very big platform. And I'm really happy and grateful like I got a chance to present in front of such a great, honorable like personalities who are like at the top of pharma industry. So I'm very thankful for the opportunity. And I also thank my professor, my supervisor who has been of great support throughout my research journey. Professor Sanyog Jain, sir. Thank you, sir. And I would also like to thank my mentors, Kaushik sir and Rohan sir, for their support and for teaching me ABC of research. And also all the lab mates and batch mates. Thank you. Thank you so much.
Is Dr. Sanyog Jain is there in the panel today, Tarun?
Yes, sir.
Yeah.
Yes,
Yeah,
sir.
he is there.
Yeah. Good evening, Dr. Sanyog. Welcome. How do you feel that when you are to one of your students winning the prize at the global level? Your feelings, please.
Cannot hear, sir.
Muted.
Is it muted? Dr. Jain?
Yes.
Please.
Am I audible now?
Yes.
Yeah. Greetings to everyone, and thank you. It's a great honor for us. So this I am associated with this SPDS since 2019 when when SPDS held the DISO India conference at
Okay, I think
from my group, they are continuous. So of course, this DRPI concept was initiated during the COVID time. So it's really it's a grand success as compared to the physical conferences. So this online conferences. So great congratulations and best wishes to the SPDS and
Thank you. Thank you, Dr. Sanyog. Maybe some disturbance in the, very nice. Thank you so much.
Yeah, thank you.
And we'll come back to you again.
Sure.
We look forward to a lot of support in the coming years to add value to SPDS and its initiatives. Once again, thank you, Darshini. Tarun, is any chance the earlier candidates, earlier winners, supervisor or guide is also present?
No, Derajaram is not there, sir. I
Okay.
couldn't find him on the panel.
Fine. So once again, Darshini, congratulations, and you can feel proud and you can work more harder. Maybe next time you can aim for the #1 position. Okay. May not be in the MPharm category, maybe in the PhD category, if you are continuing your research. Good.
Yes.
So keep in touch with us, and because I can share a great experience which has happened for me last week when we had a NIPER, all NIPERs together. There was a meeting in Hyderabad, and I was attending that PharmaCon. And there I found one of our winner like you, who won the prizes when we started DRPI, has become an assistant professor now in the university at NMIMS. And that is none other than Dr. Mukta. And it's so good to see her in that platform, and she was so happy after so many years. Like this, maybe in another platform, we will meet you also sometime later. Thank you very much. All the best once again. Yes, celebrate this victory with your parents, with your friends, and with your teachers. And NIPER Mohali, can we feel proud of you as a winner as well as a student? Okay.
Yes, sir. Thank you, sir. Definitely.
Great job.
Thank you,
well
sir.
done. Now
Thank you, sir. Thank you so
we'll
much.
go and see the winner. So Tarun, can you now unveil the winner of the Global DRPI Young Researchers presentation? The first prize winner is Asil Al-Rijebe. Taylor's University from Malaysia. Let's give a big hand. Congratulations, Asil. You have done a wonderful job. Very good presentation. We all were listening. The title of your abstract is Drug Release from a Biodegradable Chemo Radio Metabolization Agent Loaded with SM-153, Radionuclide and Doxorubicin. And the guide is Professor Chai Hong Yong. Fantastic job, very well done. Congratulations on behalf of the entire SPDS and DRPI leaders present and non-present today in this platform. Let's give him once again a big hand. The first prize is sponsored by Sotex, $2,000 you have won, and a great certificate signed by the leaders of all chairs across the world of DRPI. Tarun, you can show that. Yes, can we get Asil in the panel now?
Hi, thank you so much. Thank you. It's a great honor to be to win this, and I gained a lot of knowledge and opportunity, and I'm very happy with even my colleagues who hear all of this from different parts of the world and how we will be impacting the future with our research. So for the early stage and also for those who've done their PhD. So I can't wait so that we all can actually meet face to face. So if anybody wants to come to Malaysia, please come contact me.
Sure.
And thank you so much for all the organizers and for all the panels, because this would not have happened without you. And this was a very, very successful event, and I've gained so much knowledge. So I'm very happy, and thank you so much.
What do you plan to do, Asil?
Well, now I'm moving forward. I want to further my studies into PhD, and this project will be moving towards animal studies. So maybe you'll see me around soon.
Soon, within a month's time, many universities will call you because this news will get spread world over today. So you will have many offers from the university. Hey, come here and do PhD in our institute. So you have any choice in your mind? If you're given a chance, which one you would like to go for?
I would want to continue with the same university because of my supervisor.
Okay. Any chance your supervisor is there in the platform today in this panel?
Unfortunately, no, she is with another meeting because it's 10:30 p.m. our time, and she had to leave for another meeting online as well.
So anyway, our congratulations to her also. Keep continuing your research. Definitely Malaysia is not far. Maybe we can look at in future some events where you can collaborate with our science initiatives, make presentations. You can give a lecture or a talk. Let us see how we move forward. Once again, congratulations to you. So I request Dr. Saranjit Singh to join with me and hand over the certificate along with Sotex $2,000 cash prize.
It's my great pleasure to hand over Asil this award of $2,000 and this very coveted certificate. You have done wonderful. There was a good competition. All the small number of the participants were there. But yesterday, Dr. Samir was telling, don't call it a competition, but a motivational participation. So I think you must be very motivated, and I want to see you a future great researcher and connected with the dissolution science. Thank you. Best wishes. God bless.
Thank you so much. Thank you.
Thank you, sir, for your good words. And Asil, please continue your journey in this field of research. And at any point of time, if you feel that SPDS or Sotex can be of any help to further enhance your research or help you to do more research, feel free to be in touch with us at any point of time.
I would like to add that I will have special thanks to Dr. Matthias for conducting this JCA event, that we have the student from Malaysia winning this competition.
Do we have Matthias here? Dr. Matthias? Yes. How do you feel that the student from your zone, your region has become the #1?
I think when you just have seen the presentation that Seal has presented, then actually this really makes my heart sing, because it was awesome science. I liked the presentation very much. And now I'm very much happy for the science to basically also be now rewarded in that way. So, and the contribution of JC was really minor. I think it was Asil who really delivered here awesome science. Thank you for that.
Thank you so much. Thank you very much.
Thank you, Professor Matthias. And in fact, tremendous efforts you have put to identify the right candidate. And I'm sure next year in the PhD category also, correct? And we don't know the results. Maybe we will wait for it. So thank you, Professor Matthias, for getting this brilliant candidate research into this competition. So once again, thanks, and maybe we'll work more together next year to get more presentation from more universities in your zone, including Japan. Thank you. So we have now finished the category of young researchers, that is early stage researchers, masters and PharmD group, postdoctoral. Now we will go to the late stage researchers category, PhD, postdoctoral and industry. This is a very tough competitions and brilliant presentations we have seen yesterday. Almost 10 research presentation was there, representing from all the regions who are participating in this competition. So we will go again in the same order. We will start with the third. The second runner-up, Tarun, can you put the certificate? The name, yes, thanks. It is Sushesh Srivastava Palakruti from Texas A&M University, Kingsville, Texas, USA. Congratulations, Sushesh. You have done a wonderful job. Can we have you in the panel? Tarun, is he there?
Yes, sir. Can you hear me?
Yeah, thanks. Thanks for joining. And your abstract title was or is Development and Validation of Microdialysis Based Discriminatory and Biorelevant In Vitro Release Testing Instrument for Complex Ophthalmic Products. Your guide supervisor is Dr. Nitin B. Chabre.
Sir, I have a small correction here. He is one of the co-authors, but my supervisor is Srinath Palakurti.
Okay, we can right now correct it, and Tarun will do it, and we will write in your certificate accordingly. And we regret this inconvenience cost. And first of all, I must say, you did a wonderful job. We all heard, listened your presentation fully, and you have given, I must say, a tough job for the judges also to decide, because always when you have 10 presentation, the between third, fourth, it's all hairline difference, points, marks, half a grade. I don't know how they finally they come into a consensus. You are the third prize winner, the second runner-up. So you have done a very good job. Keep it up. Congratulations to you. Your presentation was excellent. Keep continue doing this, and we are really proud of you.
Thank you. Thank you very much. Thank you very much, Dr. Ramaswamy. And this wouldn't be possible without the support from my PI, Dr. Palakurti, and also he's our postdoc. He's our previous postdoc in our lab, Nitin Charbe. So I would like to extend my thanks to them. And of course, for the funding support from FDA and also the support from the department.
Absolutely, absolutely. I'm sure if people like you don't get funding, who else will get? So their money is well utilized, I feel, with this. You are winning this competition.
Thank you very much.
So Tarun, can we display the certificate and the cash prize? The check. Sushesh, am I spelling correctly, Sushesh?
Yes, that's correct.
Okay, good. So Sushesh Srivastava, and here is your certificate signed by all the DRPI leaders of all regions, and including the president of AAPS, president of SPDS, the global chair of DRPI, US chair DRPI, JSEA chair DRPI, and Australia and New Zealand chair of DRPI. It's a fantastic once again, and you can feel really proud. You are bringing a lot of name and fame to your own institutes and your colleagues, as well as to the university. Once again, congratulations to you. May I request now BSF again, Sadish, to join up front.
Yeah, congratulations, Sushesh. A great work and a lot of things lies ahead for you.
Thank you very much.
Along with Satish, maybe we request our our previous president, Dr. Mr. Vijay Shirsagar, also to join and hand over the certificate and prize to Sushesh. Yes, Mr. Vijay. Unmute, please.
I think I was muted. Are you able to hear me now? Yeah.
Okay, we have.
Yes, Dr. Vijay, continue.
Okay, fine. So I'm extremely pleased to hand over this certificate to the deserving candidate. And I wish him all the best for all his future endeavors and look him to contribute towards human health and its advancement. All the best.
Before we hand over, it's very important and apt to listen from Hardeep Saluja, Dr. Hardeep Saluja, who is the Vice Chair of DRPI, how you feel now of he winning the third prize. Dr. Hardeep.
Well, hello everyone again. I definitely feel very happy. Congratulations, Sushesh. I mean, he did a wonderful job. And at this point, students that are winning these awards, doesn't matter whatever part of the world. If you're winning this award, you're doing well. Make sure when you are successful, give back to the community.
Sure.
If DRPI has helped you and acknowledging your all the recognition, make sure you give back to the society, to the community. I feel very happy about it. As I said earlier, when we were starting this DRPI here in US, we were a little nervous about it, how things will go. I mean, this was untested water for us. But DRPA that was already done in India actually helped us a lot because there was a strong foundation that was already laid. And for us, it became way more convenient to conduct these in US. So thank you for this opportunity and congratulations, Sushesh, once again.
Thank you very much, Dr. Hardeep, for your kind words. Please, you also join us while handing over. Before handing over, Sushesh, I want one promise from you. And that is you will spread this DRPI competition of 2024 to at least 10 more students, research students in your campus.
Definitely. I actually have a bunch of students who are already planning on participating next year.
I'm very happy. I think I forgot to say this to the M.Pharm Masters winners, but if they are on the panel, this is a request for all the winners, an assignment for them to get at least 10 more such research presentation through each of you. Okay, thank you very much. Now you take this handover the certificate by a team of people, Dr. Hardeep, Vijay Shirsagar, and BASF, the business head of South Asia, together. Okay, and this all will reach you, Sushesh, soon.
Sure, sir. Thank you. Thanks a lot.
Yes. So thank you very much. Now, having seen the second runner-up, let's go to the first runner-up who has got the second prize. So Tarun, it's a time to show the second prize winner is Pooja Thodke from ICT Mumbai, India. Let's give a big hand. Is Pooja is there on the panel? Pooja, can you start your video? Yes.
Yes, sir.
Let me read your abstract title is amphotericin B nanoformulation Insta AMB tracking safer monomeric AMB release. And the guide is none other than Professor Padma Devaraj, who incidentally the president of SPDR. Great job, good job. Congratulations to Pooja and Dr. Padma. Yes, now we will listen from Pooja what to say on this now.
Thank you so much, sir.
Yes.
Thank you so much. SPDS organized the DRPI competition associated with the apps for providing such a wonderful platform for the PhD student and the postdoc researchers, as well as the industry people, which is to platform for to showcase their research work. And the first and foremost, I'm I would like to thankful to my research supervisor, Professor Padma Videvrajan, for believing in me and constant support and guidance. And I'm also thankful to Mr. Suhas Yegle for the guidance for the dissolution study. And also thankful to the SOTAX India Private Limited for the providing me USB 2 and USB 4 facility for completing my dissolution work. And the journey is DRPI is not a competition for me. It's a learning for my future career development. Thank you so much.
Well said, Pooja. In fact, I always tell repeatedly in all meetings, USP4 is available for any student in any part of the world to do their research. I know it's an expensive mission, but if you cannot afford, if your institute don't have it, please come to us. We will, SPDS will organize an instrument for you to do your research work. If the machine is not coming, someone else will do that work for you and give the data so that you can continue your research. You did well and your presentation was excellent. Being a second prize, first runner-up in the global level competition is phenomenal, You can feel proud, your friends and colleagues can feel proud, Dr. Padma will be feeling proud, ICT is feeling proud, and SPD is also feeling proud, and India can feel proud of you. I'm sure in the next 1 month, you will receive possibly universities calling you for the postdoc work. So you can decide now which university you want to go in US or Europe. Dr. Hardeep is already looking at it. How can I get her? Anyway, so good job done. Very well done. Congratulations once again. So let's hand over the prize. Can I request Dr. Farisad, to hand over the prize on behalf of ACG.
Congratulations.
And also we call Dr. Padma to share, being the guide and president of the society. Yes, Dr. Padma, please.
Well, I think for any teacher, an achievement of their student is something that gives us a real high. And it tells us that somewhere we have made a difference to a student. And I think being part of SPDS since the last 10 years, I'm very happy that I've been able to motivate my own students to focus in this area of dissolution for their research, because generally students love to work in formulation right from end to end, and focusing on something like this, giving it a pivotal role in their research is not a very easy task. But I'm very happy that now my research students are geared towards this. And I think Pooja's success would only be a bigger motivation for everybody else, not only in my group, but even in the Institute of Chemical Technology, to start focusing more on also these very important, subtle, but very, very relevant aspects. So thank you very much. And I think this platform that has been created is a fantastic platform. And particularly, I'm very happy to be part of the team where it has gone global also. So thank you very much, SPDS, DRPI, and well, I'm very lucky to be part of this great movement. Thank you.
Thank you, Dr. Padma. And Dr. Farisad, please hand over these prizes to the winner, Pooja. And Pooja being in India, it's easy for her to collect, or we can send fast, sure.
So congratulations, Pooja, and wish you all the very best. And congrats to you also, Padma.
Oh, thank
you.
you.
You'll get many more surprises from DRPI.
Thank you, ma'am.
Thank you so much. And the final and the last winner of the whole show, the PhD, the late researcher, postdoc group or industry group, all are waiting to see who is this young budding scientist of the world. Correct, Tarun?
Yeah, sir.
Can we show now?
There is one message from Dr. Sanyog Jain saying that the guide of Bharti, the earlier winner, that is Deraj Beniwal, is also student of Professor Padma Devarajan. So.
Oh, congratulations, congratulations. Yeah, I'm sure world over their students are there. It's great. It's great. So now coming back to the winner, which everyone is waiting for, The competition was toughest of the toughest, I would say. By viewers, we all put marks, but I'm sure by any standard of evaluation, even a person who don't know that much of dissolution evaluated, people could see from that presentation, hey, this is going to get number 1. So you can now think.
Dr. Rama, invite Dr. Vinod Pishar to do the honors.
Wait, yes, I'll do that. So coming back to that, let's first see who is that winner. Any guessing? Tarun, just play it now. And that is none other than Nilofer Salehi from Eli Lilly and Company, Indianapolis, Indiana, USA. Congratulations, Nilofer. Fantastic job, very well done. Very good presentation. I would say excellent presentation made. You have created a history in SPDS and DRPI, being the first global winner of DRPI 2023.
Thank you very much. I'm so excited to hear that, and I'm humbled. It was a learning opportunity for me to learn from all great scientists participating in this event. And thanks all the people who spent their weekend on this event, and it was my pleasure to be part of this. Thank you.
Thank you, Nilofer. And your title was Comparative Evaluation of Dissolution Performance in USP2 Setup and Alternative Stirrers and Vessel Designs, a Systematic Computational Investigation. Your guide, Professor Grigory Amidon. Great.
Yes.
You are really fortunate. And your topic is also very good. And definitely, we will help you to connect with some senior scientists who are working on hydrodynamics, and you will be able to take further your research forward.
That would be a great opportunity. Thanks a lot. And I want to thank my advisors, Gordon Amidon, Greg Amidon, and also Professor Ziff to be part of this. Also the co-authors, Dr. Bart Hens and Dr. Joseph Augustus.
Thank you once again, Nilofer. Again, my request to you will be, you will spread this news in your company as well as in your colleges where you have studied, and get at least 10 more research presenters from US next year to this competition.
Definitely. I'm still involved with University of Michigan, so I definitely invite all the students from Michigan to participate in this awesome event.
Thank you. Thank you very much. And now we hand over the prize of $2,000 as well as the certificate signed by AAPS President of SPDS and all the chairs of the regions. To hand over this, we have a great person with us, that is none other than Dr. Vinod Shah, and along with me, we hand over the certificate as well as, and also can I request our Vice Chair of US, Dr. Hardeep, to join. She is from US. Dr. Hardeep, please join with us in handing over this, please. Yes, Dr. Shah, can I have you? Dr. Shah?
You need to unmute, Dr. Shah.
Unmute, sir, please.
Yes, it is great to see that in the first DRPI international competition, the first winner, top winner is from US. Congratulations, Nilofer, for that. And just for your information, Ramsamy and the others, the mentor of Nilofer is also the president of AAPS. Actually, both Greg Amidon and Gordon Amidon, they are both the past presidents of AAPS. And it's great to see that the whole thing has been again co-sponsored with AAPS. So thank you again, Nilofer, for having a wonderful presentation and winning the first prize. Thank you.
Thank you very much.
Okay, so we are also really privileged to send this prize money to you. We will take your bank account and it will get credited in your bank account within 30 days. Okay, and the certificate will be mailed to you. You can take a printout, frame it, and display wherever you want to be. Okay. Anything else, Nilofer? Last one line you want to say?
I want to thank everyone, all the judges, also US chairs, Dr. James Pauly and Dr. Hardeep and Dr. Ramsawani for organizing this. And it was a great pleasure to be part of this event. Thank you.
Welcome. Dr. Hardeep, anything you would like to say now? The winner is from your zone, your region.
Well, Nilofer, congratulations for this. I'm actually not surprised. We heard you the first time when you presented in the US event. So your presentation was definitely amazing. Everybody loved it. So we have 2 winners from US for this event. Thank you for participating and supporting DRPI, and please continue to support DRPI in future. Congratulations again.
Thank you.
Okay, that comes to an end of this grand finale of 2023 DRPI World Finals. So before looking at what next, is that I have only 2 lines to say to all the parts. There are many students who have participated in this or in the panel. You have not lost anything. In fact, it is a great experience for all of you who has presented your research work in this platform, and we were able to bring you a different type of research, different quality of research work. And I'm sure everyone would have learned from each other. And when you pursue your research from now to further years, possibly you will adopt, imbibe from these winners what are the good things they did, which the judges have found a little above you, maybe 1 point, 2 points, 3 points above you. So if you all take like that, I feel our objective of doing this competition is achieved. So all go with happy mind. People who have not got award through this platform, you will get many more or even bigger awards in your life from various other bodies, platforms, and universities. So my congratulations once again to all those who have participated in DRPI, not only in finals, semi-finals, early rounds, and abstract submission round. So thank you so much once again, and really looking forward a great event of 2024. So shall I pass on to Professor Saranjit Singh? Anything last you wanted to say now before I handing over to Dr. Krishna Priya for vote of thanks? Okay.
A wonderful 2-day treat. I am really, really happy that it has culmination in this manner. And I can very clearly see that when we are doing Indian competitions and going to global, how tough it becomes for everyone to really try best to make presentations and compete. And congratulations to the winner. Again, I will take the words of Dr. Ramaswamy. Those who have not been able to come in the first 3 category, we might be, I will just talk to our team, that we will display the results, judgment results, and you can find how narrow they would have been. And you are all winners. You are all winners. And congratulations to everyone, all student participants, that from the very early start of preparing an abstract and coming to this stage and in grand final, successfully conducted. My thank to everybody, all the global chairs, all the global co-chairs, all the organizing committee, and 100% thanks to Dr. Krishna Priya and Mr. Tarun, of course to Dr. Ramaswamy, and I really respect the presence and the guidance provided Dr. Vinod Pisha all through this competition. Thank you, everybody.
Thank you, Professor Saranjit Singh, for a great leadership, which has resulted in today the end of the first global finals of DRPI. All those things have happened well. It's because of the great judges we had, and they evaluated so well. It was really a treat for all others by asking such an intelligent and very nice questions to people without putting any presenter into any difficulty. I could see that every presenter is answering with a smile and confidence that make this platform a different from all other competition. So let's continue the same spirit with more number of researchers coming into this platform in 2024. With this, I hand over to Krishna Priya to say a word of thanks. And thanks, Tarun. Without your full support, without your IT brain, without your full efforts, this program would not have seen the light of the day. So our sincere thanks and my personal thanks to you, Tarun. Continue doing this good job for us. Yes, Krishna Priya, here is your turn. I know Tarun want to say something, maybe he will say that. Yes, Krishna Priya, can you take over? Are you there?
Thank you, sir. Thank you. Am I audible?
Very well.
Okay. So it's my honor and privilege.
Can I say something?
Okay.
Sure. Sorry. Yes, Mr. Vaidya, please.
Yes, sir. We were talking about adding more members in the next year's program. Okay. And already Tarun has sent the abstract to most of the people. I would request all the participants who have participated in this DRPI 2023 to take the entire abstract in a USB and present it to the people whom they would like to invite as members, as a compliment to them, so that they can look at it during their resting period and then get motivated to join the membership next year, because that's the best way of inviting people into the system. We can't give hard copies to everyone, but we can always give a USB of archives that are already presented to these people, and they will be able to get motivated and get triggered into the system.
Thank you, please. Dr. Krishna Priya, you can continue.
Okay, thank you, sir. Thank you. We can only be said to be alive in those moments when our hearts are conscious of our treasures. This quote by Thornton Wilder, American playwright and novelist, exemplifies what I feel now as I express gratitude on behalf of SPDS and DRPI team to all those who have contributed in making DISO Research Presentations International 2023 a grand success. So now, let us reveal the treasures in our hearts and express gratitude. First, we'll take the acronym SPDS. And S stands for Shah, Dr. Vinod P. Shah, the Doyen of Dissolution and the guiding light of SPDS. P stands for Padma, Dr. Padma Devarajan, President SPDS and a pillar of support. D stands for the driving force of SPDS, and everybody can guess who that person is, Dr. Ramaswamy. And S finally stands for Dr. Saranjit Singh, our global chair and a stickler for perfection. You four have been the pillars of making DISO Research Presentations International have a global reach. Our salutations to these stalwarts. Now we come to the acronym DRPI. D stands for digital. Right from abstract submission to video uploading to all the evaluations and compilations of DRPI, everything was digital. And thank you, Mr. Tarun Soni of Nick Interactive, for making this journey so smooth. R stands for regions. That's how we went to DISO Research Presentations International with 5 regions, and we thank all the chairs, vice chairs, organizing and evaluation teams, including those for abstracts, video recordings, semifinals and finals of each region, for all their efforts culminating in the grand finale of these 2 days. Hoping to have more regions participating in the next DRPI. P stands for participants. Without you, we would not have had this competition. Thanking all the participants and their mentors from the bottom of our hearts. Also extending thanks to all the viewers of this global event. I stands for industry. Without the industrial support as sponsors, we would not have had any awards being handed out. The awards have been sponsored by SOTEX, ACG, and BASF, and we thank them for their continued support. I finally end by expressing gratitude to all those who have contributed in any way to this global event. Thank you and see you next year for DRPI 2024.
A standing ovation to Krishna Priya for this very innovative vote of thanks.
Thank you, madam.
Yes. And my standing ovation to all the judges.
Yes.
Absolutely.
I did it. Fantastic. Well closed. Okay, the grand finale is formally closed and now you can have chat. The recording is stopped. Tarun, stop the recording, Tarun.
Okay.